Stefano, can you? Okay, thank you.
Yes, I did it. Thank you.
So, yeah, first of all, I would like to thank you both, Stefano and Giuseppe. And, it's too elogious, Giuseppe, what you say about me. So I'm just trying to do to advance this field, but there are some much better people than me. So now I'm trying to I'm going to share the screen. So can you see my presentation now?
Yes.
It's okay. It's a full screen.
Yes, I guess so. Yes, it's okay.
Okay, so I will start. So again, Stefano and Giuseppe, I would like to thank you for this very nice invitation. As I told you before, I would have liked to be in Bologna, but I had some duties here. So it was a little bit easier for me to stay in Dijon to give my presentation from here. So today we are going to speak about biomineralization, and I will make a very general introduction to the field because I think that most of the people who follow this course are not always aware of what is behind biomineralization. So I will give first a rather long introduction. Then I will go more specifically to the macroevolution of biomineralized systems, especially with reference to the calcified systems. And then I will go more specifically to mollusk shell biomineralization. And at the end, I will finish, if I'm still alive, I will finish about some applications that we can have. So normally I should really cover the 4 hours, you know, and probably, so I prepared something which is probably more than 4 hours, but so just tell me stop when I have to stop, okay? So what we can do is maybe after 45 minutes to give to make a short stop, a short break, and then continue with the second presentation and so on. So as Giuseppe said, I work already for 19 years in Dijon, in the capital of Burgundy, and which is known as the mustard town. And I work in biogeoscience unit, which is a twinned unit between University of Burgundy and CNRS, and we are about 120 permanent people and non-permanent employees. And our big group is divided into different teams. And one of them is called Samba. This is an acronym, but I don't remember which one. And within this Samba team, there is this sub-team, which is biomineralization. And we are actually, it's a very small team because we are 2 permanent members. But this team exists already since 2003. And we used to be a little bit more, but then we have shrunk a little bit. So this is the context. And today I'm going really to give some, in this first part, in this 45 minutes or 50 minutes, I will give some really some ideas about what biomineralization are. And when we speak about biomineralization, we want to say about different things. It has different meanings. First of all, these are mineralized structures which are produced by living systems. So biomineralization are objects. But then when we speak about biomineralization, it's also a process by which living systems produce minerals. So we see it more from the physical and biochemical part, biochemical side. And finally, biomineralization is a scientific discipline in its own. And this is this aspect that I'm going to speak about now. So biomineralization, when considered as a scientific discipline, is at the crossing of many, many different disciplines. Some of them are fundamental, like crystallography, mineralogy, archaeology, paleontology and evolution, sedimentology, geochemistry, environmental sciences, physiology, biochemistry, molecular biology. And some of them are more applied, So this is, for example, aquaculture or pearl culture, biomaterials, health, bone and dental surgery. So now I'm going to give a little pinch of history of this discipline, of the field. And we can go quite far, actually, because if we look at the use of biominerals by human beings, it goes back to in the roots of prehistory. And probably among the first use of biominerals by human beings were the use of shells. And some shells collected by prehistoric population were sewed on clothes or were used as ornaments. And I give here the example of the Grimaldi children, the Cro-Magnon children of 11,000 years ago. Shells were also, shells but also teeth were used also as ornament by prehistoric population. And in the Peri-Pacific islands, shells were used as currency. We all know the cowries, which were these gastropod shell used as currency as exchange. And also in the prehistoric time, in the Neolithic time, we noticed that the first glue was made from bone and skin collagens. So we already had here an example of the use of a product which is associated to biomineralization. If we go a little bit closer to us, I show here the first dental implants which were used by Maya. That's about 1,000 years ago, and you see here in this lower jaw that three pieces, three teeth, are actually made of nacre. So they were implants made from bivalve nacre, and this was documented by Bobbio in 1972. What is very interesting is that you had a complete osseointegration into the jaw of this person. So we had here a sort of surgical use of biominerals a long time ago. Well, if you look at in the Middle Age, shells, especially shells, were really used as religious symbols. So, for example, here you see, well, it's maybe not so clear, but you see little heads of Buddha just made in the shell of some freshwater mussels. And shells were used as symbol here, like the scallop for the pilgrims of Santiago de Compostela. And also here you have this Tridachna that you can see in some churches. You know. So from the 17th century, shells were really used for marquetry work, and in lots of museums you see this kind of art, incrustation of nacre in furnitures, but also as manufactured objects like spoons and like you know, like the shell of Nautilus, which was used as a sort of glass. And of course, I mentioned here the buttons for clothes. Then it was also used as jewel. We all know about the pearls, and as I said before, the invention of the use of bone or skin collagen for making glue is very old, but the first industrial fabrication of glue from bone and skin collagen was at the end of the 17th century in Amsterdam. So another use of biominerals, which is very simple use, especially in the 19th century, but also 20th century. The soil enrichment with powder of shells. So when you crush shells, you can spread them on the soil, especially when the soils are acidic or very poor in calcium. But now let's go more to the, let's say, scientific use or scientific basis of the biomineralization as a discipline. So probably the first who described the human skeleton was Andreas Vesalius, and he dissected a body that was a guy who was condemned to death and was unfortunately killed. So his body was dissected, and then Vesalius completely reconstituted the skeleton. And this skeleton can be still seen at the Anatomical Museum of the Basel University in Switzerland. So that was in 1543. Then you have 1 century later, you have Jan Swammerdam and Anton van Leeuwenhoek, who sort of made the first optical microscope, which was around 1660. And the invention of the optical microscope was was rapidly used for looking at different objects. And for example, if you look in the Micrographia from Robert Hooke, the English scientist Robert Hooke, you can see that he made the first observation of biominerals under microscope. He speaks about gravels in urine. So he detected for the first time that sometimes in urine you can have some biominerals. And this is probably really the first microscopic observation of biominerals. Well, let's go a little bit later. Clopton Havers. Clopton Havers, a British scientist, made the first description of bone microstructure. And in his treatise called Osteologia Nova, or some new observation of the bones on the parts belonging to them, with the manner of their accretion and nutrition. So that was in 1691. Later, a French guy, de la Sonne, in 1751, made the first experiment on burning bones and see what it was composed of. Later, in a completely different manner, there were 2 French chemists, Braconnot on one side and Audier on the other side, who discovered chitin. And I mention chitin because chitin is very important in biomineralization field. I mean, chitin is used by many animals as a support of biomineralization. At the middle of the 19th century, Frémy, another chemist, French chemist, really he's probably the guy who made the first modern chemical characterization of biominerals. He did that for bone, teeth, crustacean teguments, gorgonian coral exoskeletons, and mollusk shell, nacre, but also cuttlefish bone. So it's kind of nice to read what he says, because he made it in a very naturalistic way, meaning that he says, for example, my neighbor or Mr. X or Y gave me a piece of shell, and then I just started to analyze it. I just tried to dissolve it with different solvent, with alkali, and so on. And that's how he really the first guy who characterized the nacre matrix and give a name for that. Huh? So if we go to the second half of the 19th century or the first half of the 20th century, several chemical analyses of biominerals were performed, and several observations with optical microscope were also performed, especially by Schmidt, the German guy in the 1920s, and also Bogild in the early 30s, late 20s, early 30s. And Bogild is known especially for a treatise that he made on the shell microstructure, which was really the first extensive treatise on mollusk shell microstructure. So let's go a little bit further and look at the after-war period. After-war period is marked by, of course, the huge development of biochemistry and the development of several techniques for separating biomolecules. Especially at that time in the 50s, amino acid analysis started, and a lot of people started to analyze bone tissue, teeth, shell, and so on. So in this short period of 1953 to 1955 or 56. In parallel to this biochemical development, physics also was used. And for example, XRD of biomolecules. For example, in the 50s, Ramachandran, an Indian researcher, found the 3D structure of bone collagen. And you know that the bone collagen is a triple helix, very complex structure. You have the double helix of DNA and you have the triple helix of collagen. So Ramachandran discovered that. In the 60s, one can say that several biochemical characterization of biominerals were performed, and also the electron microscopy like TEM and SEM continued to develop. So many observations were made and published. And the early 70s was marked by the first international symposium on biomineralization in 1970. Now, if you look now, some months ago, we had the 16th symposium, which was in China in 2021. So that was the 16th edition. And also in the 70s, we had the first molecular models on biomineralization. I will speak about that later. So in the 70s also, a lot of big names emerged in this discipline, such as Heinz Lovenstam, who was probably the first guy who made a very extensive analysis of all biominerals found in the natural systems, from bacteria until vertebrates. But you have also Kenneth Simkis, Art Weiss, Arthur Weiss, Melvin Glimcher, Wolfi Traub, Bill Landis, Abdus Saluddin, Janet Oldijk, Carl Wilbur, Steve Weiner, Harry Mudvei, Miles Crenshaw, Peter Westbrook, Lee Haddadi, and so on. So I just want to refer here to two fundamental books in the field. So the one of Lovenstam and Weiner on biomineralization, published in 1989, but also at the same date, during the same year, the one of Simkis and Wilbur, which is also called Biomineralization, Cell Biology and Mineral Deposition. So these are the two Bibles, I would say, of people who do biomineralization studies. Today, the discipline is very active. Huh? And beside the International Symposium on Biomineralization, we have several important scientific events, such as the Gordon Research Conference on Biomineralization, which takes place every 2 years, every 2 per years. And also the ICCBMT, the International Conference on the Chemistry and Biology of Mineralized Tissue. But beside these events which are strictly focused on biomineralization, we also have several international or national conferences which have sessions dedicated to biomineralization, such as the Goldschmidt Conference, the International Marine Biotech Conference, International Sclerochronology Conference, and so on. So many conferences have like 1 or 2 sessions devoted to biominerals. So this, I gave here a very short summary of the discipline biomineralization. So now we are going to see a little bit more in detail what biomineralization is. So as you can imagine, it's a widespread phenomenon because it goes from bacteria to vertebrates. And it is about 55 phylums, living or fossil phylums, both in bacteria, in archaeobacteria, but also in eukaryotic systems. And Lovenstam, I mentioned him already, but Lovenstam was probably the first who really made a very precise inventory about all the different minerals that were formed by all these systems. And he found that there were about 6, at that time when he published his 1981 paper, I think he was about 65 minerals. Since that period, a few more minerals were found. But so it means that there is a diversity of the mineral formed by organisms, but also if you look at the amount of minerals produced by purely abiotic systems, like volcanoes and so on, we have more than 3,000. We have between 3,000 and 4,000 minerals. So 70 is not so much actually. Huh? And these minerals, they enter into different categories, which are illustrated here. First, we have the carbonates, and I will mostly focus later my talk on carbonates. But we have also silica. Silica is also a very important mineral formed by organisms, such as diatoms, such as silicoflagellates, or radiolarians. You have phosphates. Phosphates are, of course, we are concerned by phosphates because our body, our skeleton is made of calcium phosphate. Our teeth too. So beside these three minerals or three categories of minerals, we have halides, which are very, very restricted actually. Some few gastropods only have developed some plates that are made of calcium fluoride, so which is very, very rare. Beside that, we have also organic minerals. It can be strange to speak about organic minerals, but they exist. Actually, we make organic minerals when we have problems with our kidneys. So some of the kidney stones are purely organic, and they are usually made of anhydrous uric acid. And uric acid is really an organic molecule. You have sulfates. Sulfates are also rather rare in the biological systems. To give an example, some cnidarians, some jellyfishes produce calcium sulfate just as little organ for equilibrating their umbrella. So these are called statolics. So besides sulfates, we have also oxalates. Oxalates are also rather rare as biominerals, but they can be found as pathological mineralization in kidneys. Calcium oxalates are actually the most current, the most common pathological mineralization in the kidneys. Beside the oxalates, we have the sulfides. Sulfides are made by bacteria. We have also manganese oxides made by bacteria, and we have iron oxides. Iron oxides are also produced by some bacteria, but also by some mollusks, like the chiton. Chiton can produce in his organ for grazing rocks. He produces a little bit of iron oxides. So in the field, it's rather common to sort of distinguish two types of biomineralization. Nowadays, this dichotomy view is a little bit blurred, but still it's good to keep this in mind because it gives the feeling that there are two families of processes for making minerals. In the first part, there are what we call the biologically induced mineralization, and second, biologically controlled. So I'm going to just speak a little bit about biologically induced mineralization, and then I will focus more on biologically controlled ones. So biologically induced mineralization is usually defined as, because it does not require very specific macromolecular machinery, and of course this definition should be probably tempered a little bit. But the formed crystals, the crystals that are formed under a biologically induced regime, They really look like chemically precipitated crystals. Biologically induced mineralization depend rather strongly on environmental conditions. And usually they are defined by the fact that there is no real control on the shape and layout of the crystals. So which kind of organisms produce biologically induced mineralization? Well, you have a large bunch of bacteria, fungi, protists, algae, and also pathological mineralization in metazoans. A well-known example are the stromatolites, and I will come back on this when I will speak about evolution of biomineralization. Stromatolites are, okay, you have here a fossil version of stromatolites, and here you have some living version of stromatolites in Turkey Lake, in Salda Lake, but also in the Bahamas. And the stromatolites are very interesting because they form by microbial communities that make biofilms, and these biofilms usually make different layers. For example, on the picture here on the left, you see a biofilm, section of a biofilm, and you can see that you can see different colors, and these colors correspond to different type of bacteria. So the green one corresponds to the phototrophic bacteria, so cyanobacteria. The, let's say, yellowish, or not yellow, pinkish one correspond to heterotrophic aerobic bacteria, and the black one to heterotrophic anaerobic bacteria. So all these communities are really structured in biofilms, and these biofilms produce these deposition of calcium carbonate, such as you can see on the right side here and here. Well, stromatolites are, of course, they are the oldest biomineralized construction made on Earth, because they date about 3.5 billion years. And there are lots of examples in Australia and South Africa. And I will come back later to this. Again, some modern stromatolites. And let's jump into biologically controlled mineralization. So biologically controlled mineralization, they require a lot of things. First of all, they require very specific molecular and cellular machinery, and we are going to see together what it means. Biologically controlled mineralization are always made in a confined space. So it means that there is really what people call a space delineation, so a space delimitation, so where crystallization takes place. And different organisms have found different strategies for making that. As a third item, the form minerals are usually very different from their chemical counterparts. And we will see a couple of examples of that. Usually, this type of biologically controlled mineralization, they imply a multi-scale organization. Number 5, they are far less dependent on environmental conditions than biologically induced mineralization, which means that biologically controlled mineralization can take place even when they are not favored by the environment. And number 6, the mineral deposition is highly controlled by an organic matrix. So what organisms use biologically controlled mineralization? First, you have magnetotactic bacteria. We will briefly see this example. You have all kinds of protists, so single-celled eukaryotic organisms. You have algae, and of course, metazoans, so pluricellular animals. So let's see together. I will go into detail into these 6 different points. So, point number 1: very specific molecular and cellular machinery. It means that you need specialized cells. For bone, you have osteoblasts, osteocytes, osteoclasts. So these are osteoblasts are the bone-forming cells. The osteoclasts are the bone-destroying cells. These cells are also the cells are usually specialized in organs, and the typical example is the mollusk mantle that you can see here. Well, on the border of the shell is made it's made by this mantle. We will see it better later. And this kind of organ appears very early during the development of animals. So it means that upstream the organ itself, there is a complete gene regulatory network which will assign cells specific functions. So biomineralization then appears as a very integrated system within the organism. Again, point 1. Let's go more in detail to the mollusk mantle. And I just show here an oyster that I have opened for Christmas time before eating it with good French white wine. And before doing that, I just took this picture. And you can see here that the oyster has its calcifying mantle. So this thin organ is the one which made this very thick shell. And it is believed, well, there is no real proof about that, but the mantle will produce, first will extrude the mineral components, the ionic precursors of mineralization, and it will also extrude the organic components. Point number 2, space delineation. And the first nice example is about the coccolithophore algae. Here you see Emiliania huxleyi, which is one of the most common coccolithophore algae in the ocean of today. And each of these cells is a single cell, and it produces coccoliths. So coccoliths are these little rounded plates which are made of calcite, which are actually made inside the cell and then exported to the outside of the cell. So there is this drawing showing that if you make a section in the coccolith algae, so you see the nucleus, the chloroplast. Here you see a vesicle which is fed by the Golgi apparatus, and the vesicle will accept some components, and in this vesicle, which is really attached to the nucleus, you have the secretion of a first template, I would say. This template gets mineralized progressively, and after a while, the vesicle detached from the nucleus and will migrate to the external of the cell. And here, each coccolith is really made in one vesicle. So again, here you have a delineation of the space for making the coccolith. If you look now the mollusk example, the strategy is quite different because mollusks do not use a vesicle, although it can sometimes use vesicles, but the strategy is slightly different. Here you have a mollusk. If you make a section across this mollusk, this bivalve, you will see that, so you will see the shell here, you will see the mantle here, which is making some folds. One of the folds of the mantle will produce, you have specialized cells here which will produce what we call the periostracum. And the periostracum is organic, very deeply insoluble substance, extracellular production, which will seal the space, the very enclosed space here, between the shell and the living tissue, the mantle. And because this space is enclosed, you can the mantle can extrude the mineral precursors, the mineral ion precursors, and also the skeletal matrix, the shell matrix, which will be interact with the mineral precursor and allow calcification to happen. So here you see in this graph that the mollusks have invented somehow the perostracum, so these organic layer which seal a space which is not in direct contact with the environment. Point number 3, the foam crystals made by biologically controlled systems, the foam crystals are usually different from their chemical counterparts. And there is this very well-known example of calcite. So here on the left, you have a crystal which is made purely abiotically. It's made when you let diffuse ammonium bicarbonate into a solution of calcium chloride. You get these very beautiful rhombohedrons, which are typical calcite rhombohedrons. Here, so this is a monocrystal. Here you have the spine of sea urchins. If you look at the spine of a sea urchin, it looks very complex. You can here you have this little needle. When you zoom, you will see that this needle is has some substructure. You zoom again, you see that it's very delicately sculpted. But if you do an X-ray diffraction on one spine, it will just behave as a monocrystal, similarly to the abiotic calcite crystal. So this is all the difference between biologically controlled mineralization and abiotic mineralization, meaning that abiotic mineralization, sorry, meaning that the organism itself sculpts its own production. Point number 4, multiscale organization. And there is the well-known example of the vertebrate bone. In the bone, one has found at least 7 levels of hierarchy, from the nanometer to the centimeter scales. So everything starts from the collagen protein. Collagen, the unit of collagen is one peptide, one very long peptide, which actually is organized in a triple helix that you can see here and here. The triple helix will organize itself in different, let's say, arrays, with a little shift between these columns of collagen molecules. And then this organization is then multiplied somehow, forming mineralized fibrils. These fibrils made bundles of fibers, which themselves made lamella. And at the end, you have an osteon, so one unit of the bone. And at the end, you have the bone. So you have here, really here, a complete organization in different hierarchical levels. We could say, well, this is because vertebrates are evolved organisms. But if you look at metazoans, which are supposed to be more simple, you see that it's also the same. Meaning that here we have the red coral, which is this precious coral you use in jewelry. Red coral is the Corallium rubrum. It's made of magnesium calcite. And it has also different levels of hierarchy, because you take one of these little branches that you cut, you will see this structure. So each of these, let's say, elements are composed of what we call herringbone units. And the herringbone units are made of spindles here. The spindles are themselves made of sort of lozenges, which are also made of little fibers, and the fibers are made of subunits and so on. So we go here from the, let's say, millimetric scale to the nanometric scale. And this system was really well studied by Daniel Vielzeuf in Marseille. He published about a decade ago all the structure of this corallium reborn. So again, biologically controlled mineralization are usually present a multi-scale organization. Biologically controlled mineralization, and that's the point number 5, they are far less dependent on environmental conditions than biologically induced mineralization. And we have a beautiful example which is that of acantharians. Acantharians are really planktonic marine protists, so single-cell animals, which produce a little sort of skeleton, we call it a test. And this skeleton, it's not made of calcium carbonate, it's not made of silica, it's made of strontium sulfate, of celestite. And strontium sulfate is a very, very unsaturated in marine environment. So it means that it's very highly unstable, meaning that when the animal dies, the skeleton will float and then go down and then be dissolved. So it means that the animal probably use a big part of its energetic budget in making such a skeleton, such a test of strontium sulfate. And of course, there is no reason why it does that. We don't know why it does that, but it does it. Other examples taking from Metazoans. So again, here you have the example of the freshwater mussel. You can find it in the acidic water, which are usually undersaturated in calcium carbonate. And these mussels, these freshwater mussels, they can live very long. They have a growth rate of their shell which is very, very slow. And one speaks about shells that are as old as 80 or 85 years, meaning that they have a very low metabolism. They produce calcium carbonate at a very, very low pace, but they can live very long. Another example is that of the Ascidian, Erdmania momus. Erdmania momus is an Ascidian. Ascidians are defined as urochordates, so they are sort of cousins of vertebrates, so to speak. And urochordates are Ascidians they look like bags with exhaling siphon, inhaling siphon. They are not very impressive, but they are sort of cousins of us. And some of them, like the Erdmania momus species, produce spicules, and the spicules are not calcite or aragonite, but they are made of vaterite. And vaterite is a very unstable form of calcium carbonate. And usually vaterite is not stable in the in the natural environment, but it will automatically recrystallize either in aragonite or calcite, or be dissolved. So again, it means that in biologically controlled systems, animals can reproduce minerals which are thermodynamically unstable. And the last point, biologically controlled systems use a very complex organic matrix for deposition of the mineralization. I here let's say, illustrate this by a few examples. You have here the diatom, Thalassosura, Syrah, Pseudonana. These diatoms use a lot of proteins, and not only proteins, but also long-chain polyamines, just for the deposition of their frustule, their silicified frustule. You have demon sponges. Demon sponges also use a lot of different proteins like silicateins, galactins, collagens, selenoproteins, and so on, silicases, and so on, for depositing their siliceous spicules. Sea urchins, and one of the most known models is Strongylocentrotus purpuratus, which has been studied for now 30 years. These animals use a complete repertoire of different proteins, some of which are listed here, for the deposition of its spine and its skeleton. Mollusks, of course. Mollusks use a lot of different proteins, and we will see that later in the second part of my talk. And of course, chordates. Here, for example, the tooth of vertebrates of human beings. They are, let's say, deposited owing to a complete cortege of proteins, including MAPE, amylogenin, amyloblastin, enamelin, amylotin, and so on and so on. And all these processes are also controlled not only by proteins, but also by other components like sugars, like polysaccharides. This is what I show here. PTM means post-transactional modifications, meaning that many of these proteins that I symbolize here, that I list here, exhibit post-transactional modifications such as glycosylation or phosphorylation, and these also allow the deposition of minerals. So we can go on now quickly and look at different examples of biologically controlled mineralization. First of all, let's see a bit magnetotactic bacteria. You know, magnetotactic bacteria are the bacteria which can orientate according to the magnetic field, the Earth's magnetic field. And nobody knows exactly why they do that, but they do. And they produce actually magnetite nanograins from 25 to 100 nanometers, which are synthesized by an organite, which is called a magnetosome. And this process has been really well studied by some German colleagues, and they have completely identified a different set of proteins that are used for controlling the deposition of these magnetite grains. And most of these proteins, maybe not all, but most of these proteins, they belong to what they call a MAM family. And what is nice is that they are able to, by switching some gene off and on, modify the shapes of the magnetite grains. Another example of controlled mineralization, which was published about 8 or 9 years ago by a French team. Usually, people believe that bacteria are mostly doing biologically induced mineralization, but not biologically controlled. And this is especially the case for calcium carbonate minerals. And here was published the first example of a bacteria which produce calcium nodules, intracellular calcium nodules. And it does it probably in a very controlled way. Let's say the chemistry behind is still not well understood, but this was the first example of bacteria producing controlled calcium carbonate nodule. Well, again, another example of the controlled mineralization, of course, the coccoliths. I will not go into detail. Diatoms, well, for diatoms, silica is produced. And diatoms is a huge group of organisms, of algae that appeared about in the Triassic period, about 200 million years ago, and they started to know a big development in the Mesozoic time, so Jurassic and Cretaceous, and also in the Cenozoic, meaning in the Tertiary era. And diatoms colonized freshwater and marine waters, and they are really key players in the regulation of silica at the global scale. Another example are the Foraminifera. Foraminiferas, are protozoans, little single-celled organisms that are able to produce the test of a little skeleton, which is usually made of calcium carbonate, which can be calcite or aragonite. And they were extraordinarily successful in the Cenozoic times, and they are still very successful. I mean, if you walk on the sand on the beach, everywhere you have these single-cell animals that produce their little test. So they contribute also a lot in the global production of calcium carbonate from a biological system. Another example are the sponges. So the sponges, also called poriferans, they are able to handle two mineralogies, either calcium carbonate, and this is the class of calcareous sponges. So they make spicules, like needles of calcium carbonate, very often calcite, but sometimes aragonite. But they can also produce a silica-based skeleton, like this animal. This animal, it's a glass sponge. It's an hexactinale sponge. So beside the hexactinale, some other types of sponge called a demosponge can also produce silica-based spicules. And even in the group of demosponge, some of them, few of them, are able to produce silica-based skeleton plus calcium carbonate-based skeleton. So you can find both skeleton together. So that's only a little group of demosponge who can make that. But they can deal with the two completely different mineralogies and chemistry. Another example are the cnidarians or coelenterates. So they can produce, of course, a huge amount of calcium carbonate, and I show here an atoll in the French Polynesia, where all these atoll and what is below is completely made of biogenic calcium carbonates. They make these huge reef systems. Cnidarians are also able to deal with two mineralogies, let's say, within calcium carbonate. They can use what we call two polymorphs of calcium carbonate. Octocorals, like gorgons or like red coral, they use usually calcite. They use calcite. While scleractinian corals, so the corals which make most of the reef systems, they use aragonite. So both are polymorphs of calcium carbonate. Calcite is the rhombohedral form of calcium carbonate, while aragonite is the orthorhombic form of calcium carbonate. Another group of organisms, Brachiopods and Bryozoans. So brachiopods, they look a little bit like a mollusk, but they are not mollusks at all. And it's a completely different phylum. They produce a shell which has two valves also. And usually this shell is made of calcite. There is one big exception. Within the group of brachiopods, you have lingulid brachiopods, which have a shell made of calcium phosphate. And this is the single exception to that. Beside the brachiopods, you have bryozoans. Bryozoans are colonial organisms, very tiny. They are also reef builders, although their role is a little bit below below that of corals. But they also produce an exoskeleton, which is usually made of calcite, and sometimes some form make aragonite skeletons. We have another example, which is quite funny and a little bit different from the others. These are the examples of the arthropods. And many arthropods, like crabs and lobster, shrimps, and so on, they of course, they have a cuticle. They have an exoskeleton, which is a chitinous exoskeleton, and a crayfish also. This is a crayfish. They have an exoskeleton, but this exoskeleton is mineralized, so it is hardened by calcium carbonate. And beside this exoskeleton, they have what people call gastrolites. So meaning that if you cut the head of this crayfish here, or of a lobster, you will find some these structure, these sort of concretions called gastrolites. And gastrolites are calcium storage structure. They are not made of aragonite, not made of calcite, but made of amorphous calcium carbonate. And I later on, I can go a little bit further with this model. Again, in the arthropods, you have this animal, which is an amphipode. And this amphipode has also a cuticle, which is mineralized, which is lightly mineralized. And if you made a radioscopy of this animal, you will see that inside, as a, let's say, parallel to the digestive tube, you have sort of concretion of calcium carbonate. And these concretions are actually used as a calcium storage structure. And let's say what makes arthropod calcium carbonate mineralization a bit unique is the fact that they are molting. To grow in size, they have to molt. They have to change their cuticle. So there is a complete cycle of molting, which involve hormones and so on. And during the molting process, which is called also ecdysis, they actually so they change their skin, and their new skin, which is completely soft, has to be hardened, especially by mineralization. So to do that in a short time, they have to use reserves, calcium reserves or calcium carbonate reserves from different structure. And for this animal, this is the structure that is used as a reserve of calcium carbonate. So just before the mold, the tube is full and then well made, and after the molting process, you see that all these grains are dissolved. So all the calcium, not all the calcium, but a part of the calcium from this structure is dissolved and then is driven to the cuticle for the hardening process. And this is the same for let's say hardening the cuticle of crayfishes. Well, another example of the biologically controlled mineralization, this is the mollusk shell. So here you have different examples. You have the pearl oyster, snail, and gastropod, and nautilus. But we will speak about that later. Just to show here that the mollusk shell is an important, mollusks are important contributors to the formation of calcium carbonate at global scale. Here, I see a thanatocyanosis, so an accumulation of dead shells. And here, you see a complete reef, a fossil reef made only of specialized bivalves. And I will come back to this later. And finally, the vertebrate is also, of course, one of the biologically controlled mineralization system with the bone and the teeth. So I have finished for the first presentation. We can go to the next one. Can you see it?
Yes.
Okay. So what do you want me to do? Shall I make a little break or shall I continue?
I suggest 5 minutes break.
Okay, 5 minutes, because this one is long too. This one will take 1 hour at least.
Okay, so please, 5 minutes. I ask the students to be on time, just for have a cup of coffee and go bathroom. 5 minutes, we restart. Thank you.
Okay, good.
Okay, Mariana, we can restart if you call the students in, please.
Okay.
Thank you.
Everybody's arriving.
Okay.
Okay, we're here.
Okay, shall I start again?
Yeah.
So everybody sees my screen?
Yes.
Okay. So in the second part, we will focus a little bit more on about marine biocalcification and its origin and evolution. So as I said here, so this is one of the slides of the first presentation. Biominerals are extremely diverse, and among this diversity, the carbonates have a major role, plays a major role because they are the most produced minerals at the surface of the Earth, together with silica. And as a preamble, if you look at the surface of the surface seawater, it's highly supersaturated to respect to calcium carbonate, with respect to calcium carbonate. However, places where spontaneous calcium carbonate precipitation occurs are relatively rare, and you can see that, for example, in Bahamas. And actually, in the seawater, you have several inhibitors of precipitation, such as magnesium ion, or organic long polymers like polysaccharides or even proteins or peptides. And you have also some other molecules such as phosphates or citrates. So this is something to keep in mind. Let's look at the calcium carbonate biomineralization and the carbonate cycle. So let's try to see what is the yearly production of marine calcium carbonate or calcium carbonate at the surface of the Earth produced by a biological system. So in total, it is said that between 5 and 6 billion tons a year of biogenic calcium carbonate are produced, which is, of course, quite enormous. A part of this production is redissolved, especially in the open oceans, because this when calcium carbonate from coccoliths, for example, are produced, and when the algae die, the calcium carbonate will go down, and then when it reaches the compensation line of the carbonates, it will dissolve. So this calcium carbonate will be reinjected into the system. But from above, beside these 40% which are redissolved, you have 60% which accumulates, and that represents about 3 billion tons a year. In this system, I don't speak about the continental carbonates, which are not taken into account. Now let's go a little bit more in detail. You have here the epicontinental platforms, which are domains, large domains around continents. And in the epicontinental platforms, you have an estimated production of about 2.5 billion tons a year of calcium carbonate produced. And this production is mostly benthic. This production of biogenic calcium carbonate is aragonitic, calcitic and usually high magnesium calcite. And this is made by corals, Foraminifera, mollusks, bryozoan, red algae, green algae, mostly, and also echinoderms and so on. Now we have the open ocean. In the open ocean, you have a production which is estimated to be more or less similar to that of epicontinental platform. This production is only planktonic, and it's made a big part of calcite due to coccolithophore algae, which are the main producer. But you also have aragonite from planktonic foraminifera and from what we call pteropods, meaning planktonic gastropods, tiny gastropods which actually contribute quite a lot to the global production of calcium carbonate. And beside that, we have the continents, for which the production is very difficult to guess for different reasons. This production is mainly due to bacteria, fungi, plants, mollusks in lacustrine domain. So in total, keep in mind that the marine production is about from 5 to 6 billion tons a year. Yeah, the special case of the continental domain, the production actually is highly variable because it can be very important in lacustrine domains. In some lakes, you have a huge production of calcium carbonate of biogenic origin, but it's almost zero, reduced to zero in a hot desert. In temperate forest, a lot of microorganisms, bacteria and fungi, produce important quantity of calcium carbonate, but very poorly quantified. You have, for example, the calcite in needles, which may be produced by bacteria and fungi, but its estimation is just out of our reach. In tropical areas, this production of biogenic calcium carbonate can be locally very important. We have the example of the Iroko tree, which are trees which can produce up to, each tree can produce up to 5 to 10 tons of calcium carbonate in its roots. So the calcifying regime that I described here, was it always the same across the geological time? And of course, the answer is no. And we are going to see together what happened in the different phase of Earth history. I just symbolize here let's say the geological time scale and Rhean scale, meaning that the Phanerozoic times, which start in the Cambrian until now, they just represent about 1/7 of the whole geological history of the Earth. Okay, and this long period that preceded the Phanerozoic, it's called the Precambrian, of course, which is subdivided in Hadean, Archean, and Proterozoic. And you had different events occurring during the Precambrian. While in the Hadean times, no life, only what could be guessed was that there was a prebiotic evolution. The earliest forms of life started mark actually the beginning of the Archean. The first eukaryotic system marked the beginning of the Proterozoic, and you have different events which we are going to detail, to go more into detail. So in the Hadean time, no life yet, no biomineralization. But we had the synthesis of organic macromolecule in what people define as the primitive soup. And these organic macromolecules, like proteinoids, peptides, and other components, they may have reacted with mineral surfaces, which were iron sulfide, silicates. And we probably have here, during the Hadean times, the first organomineralization and the first selection of polymers by mineral surfaces. So this selection must have been quite an important process at that time. After the Hadean, after this prebiotic evolution, life emerged between 3.5 or 3.8 billion years ago. And very quickly, what made cyanobacteria were able to construct these organosedimentary construction, stromatolites. And stromatolites really appeared during the Archean time, and they are still existent nowadays. And they knew a huge development during the Archean time and the Proterozoic. And at the end of the Proterozoic, they started to be more discrete, to be a little bit less present in the systems, because they were probably they were more efficient competitors for making biomineralized constructions. And a lot of the process of these biologically induced mineralization made by cyanobacteria, so stromatolites, this was made possible due to the formation of extracellular polymeric substances or extracellular polymeric mucilage, things that people define as EPS. And again, I show here these stromatolites, these 3.5 billion year old mineralization from the Archean. Today's stromatolites, which look exactly the same as their fossil counterparts from the Archean times. What is very important is that cyanobacteria, by making these huge constructions, which were like reefs, actually, in the Archean and the Proterozoic, they produced a lot of photosynthesis, so they completely modify the atmosphere of the Earth at that time. And they were the main contributors, or the main contributors to the formation of an oxidizing atmosphere on the Earth. I just symbolize here what happened during the, let's say, Paleoproterozoic, Mesoproterozoic, and Neoproterozoic. And at the beginning of the Proterozoic, so the Paleoproterozoic, we had the great oxygenation event. And if you look on this curve, you see that the level of oxygen, the partial pressure of oxygen is increasing at the beginning of the Proterozoic, and this is the great oxidation event. And it goes to a sort of maximum, and then it rises again close to the Phanerozoic times. And the great oxidation event had direct consequences on some microorganisms. Many of the microorganisms that were living as anaerobic systems, they just disappeared. So far, the function of stromatolites for cyanobacteria is a little bit obscure, but we can imagine that it significantly brought a certain evolutionary advantage, because stromatolites are really giving a support. They are really support for bacterial communities. They allow the structuring of bacterial communities, and I showed you before that these bacterial communities were organized in cyanobacteria, heterotrophic aerobic, heterotrophic anaerobic strains. So having a stromatolite is a way to structure the communities, but it's also a good protection against desiccation, especially if you have tides. Being on a stromatolite is probably easier for bacteria to survive. And it's also a good protection against UV light. So we can guess that stromatolites gave a really an important evolutionary advantage. If we try to think what stromatolites required, first, one of the prerequisites was the handling of intracellular calcium fluxes. You know that calcium, when considered as an ion, is a second messenger, so it's involved in signaling, intracellular signaling. And we can imagine that before mineralizing, these microorganisms had to set up a complete cytoplasmic regulatory machinery, which was also shared by everybody, because everybody needs this regulatory machinery, especially for calcium. And from that, they were able to produce their mineralization. And for stromatolites, there is the fundamental role of EPS, of exopolymeric substances, because they are the one, so these are usually polysaccharides, or a mixture of polysaccharides and some proteins, but very poorly defined, actually, a bit randomly organized. But they provide a peculiar microenvironment, which probably allows nucleation or templating of calcium carbonate. And they can play the reverse. They can also inhibit mineralization. And of course, they are not really specialized. Huh? And this is probably the big difference between the biologically induced mineralization, as it was made in the Archean and Proteozoic. The big difference between this type of mineralization and what comes later, which is the biologically controlled mineralization, which requires this complex and specialized mineralizing matrix. Let's go to the Proterozoic. So we are still in the Precambrian. At the end of the Precambrian, we had multicellular eukaryotic life. And these animals, we don't know exactly what they are. So they were published a couple of 3 years ago. They probably use chitin. And chitin is probably one of the oldest polymers that the eukaryotic systems invented. But they are not really mineralized. Huh? Then, around 810 million years ago, in Yukon, Canada, one sees the very first complex biomineralized forms of life. And this is a paper by Cohen and et al. In 5 years ago. And they seem to be eukaryotic cells. The mineral which is produced is a hydroxyapatite, so calcium phosphate. And these remains were nicely dated by rhenium and osmium isotopes. So this is probably the first, very first complex mineralization from, let's say, eukaryotic systems. Now, if we take again our geological time scale, we are going to look here, what happens here, just before what people call the Cambrian Explosion. Huh? And again, the Cambrian Explosion and what happened just at that limit is really the accession of the biologically controlled mineralization. So this is what I say, biologically controlled mineralization inherits the Earth. And this appearance of controlled mineralization in Metazoans is just one aspect of the Cambrian Explosion. But let's go a little bit before the Cambrian Explosion to see in detail what happened. If you go around 600 million years ago, you see you have the fossil embryos for Duchampion in China, which were discovered about 25 years ago. Then we find some very poorly mineralized form like Ludina just before the limit between Precambrian and Cambrian. And then from that limit from 542, 543 million years ago starts the real, let's say, Cambrian explosion and massive appearance of skeletons, exoskeletons. So first, the Dushantuo Formation in China, where you had the first fossil embryos that were remarkably preserved in phosphatic deposits. And these embryos were let's say, described by, in 1988 by Andy Knoll and some Chinese researchers. They are dated from 590 to 565 million years. They are just before or contemporary of the Ajakara Fauna, and we are just going to see later what it is. We don't know yet if these embryos correspond to macroalgae, cnidarians, sponges, bilaterians, adult metazoans. So there is no complete, as far as I know, there is no complete, it's not yet clarified. But what is funny is that these cells, they are not calcified, but they were re-preserved in phosphatic deposits. So it's very beautiful. And then, so before the Precambrian-Cambrian limit, we have the Ajakara fauna. And this fauna was discovered in 1946 in the Ajakara Hills in Australia. And it represents a very complex assemblage of macroorganisms of different shapes. And you see some of them here. And here, for example, people believe that this Kimberella shape is probably a primitive mollusk. And the main common denominator of this fauna is that they were not calcifying. They had no shell, no biomineralization, they were all soft-bodied animals. So they were remarkably preserved in the sediments. Let's list some features of this fauna. So the Ajakara fauna has a worldwide distribution, more than 30 sites on the 5 continents. It is, as I said before, it's a soft-bodied fauna, completely unmineralized. It's probably microphage, which implies a very short trophic chain. It's probably adapted to a low level of oxygen because these organisms are rather flat, which means that they had a high surface exchanges. They increased in diversity, let's say from 570 to 545 million years ago. And there would be around 70 genera and 100 species. So many of these animals look like cnidarians, like meduses, like jellyfishes, sorry. They are flatworms, arthropod-like animals, and also unknown phylum. So people say that these Ajakaran fauna may have emerged after one of the glaciations that occurred, the Gastier glaciation that occurred at 580 million years ago. And the deglaciation would have provoked an increase of the oxygenation and the rise of animals. But things are not very clear yet. This Ajakaran fauna is still very controversial. The fact that some people still think that this fauna has nothing to do with the Metazoans that appeared later, and they call the Ajakaran fauna Vandobionts, and they say that Ajakaran fauna is an aborted experiment without offsprings. This is the position of Zeilacher, for example. But most of the people say that the Ajakaran fauna is a rather a mixture of representative of disappeared and living phylums, because one may recognize in these faunas maybe some sponges, cnidarian, flatworms, arthropods, echinoderms, and so on, and also even mollusks. So we have the case of the Dickinsonia. A few years ago was published a paper showing that lipidic biomarkers were associated to Dickinsonia, and these cholesteroids seem to be typical of metazoans. So it means that with this argument, animals from the Ajakara fauna would be early representative of phylum that we know nowadays. Beside the Ajakara fauna, and just before the Precambrian-Cambrian transition, in the sediments, one can find little conical objects which were described for the first time by Germs in 1972, who gave the name Cludina, which have a world distribution, and they are really formed by calcified nested cones, so which are more or less contemporary of the Ajakara fauna, and which disappeared at the Precambrian-Cambrian transition. And we have here among the earliest mineralized or calcified forms in the late Precambrian systems. Since the initial discovery of germs in 1972, other mineralized forms similar to Cludina were discovered, like Namacalatus, Namapoikia, Cynotubulites. So there is now a complete bunch of organisms that started to make minerals just before the Precambrian-Cambrian transition. What is also nice is that one has found the first traces of predation on Cludina skeletons, and this was also published some years ago, 20 years ago or more. Now let's jump to the Lower Cambrian. What happens? During the Lower Cambrians, you have the first fauna which are there, which are really mineralized. This fauna is called the small shelly fossils of the Lower Cambrian. They are made, they have a world distribution, they have a conical shape, they're made of plates, sclerite, tubes, and they probably represent the first diversification of skeleton-bearing organisms. And one count about 40 different genera for this fauna. Some of them have, let's say, attested affinity, like Elsionella. Some of other are of rather uncertain affinities, like some sponge, cnidarian, and mollusks. More or less at the same period, so Lower Cambrian, and at the middle of the Lower Cambrian, to be more precise, some group of organisms appear, among which this animal, which is archaeocete. Archaeocetes are nowadays considered as sponges, sort of very weird sponges, because they have both, they have some characters of sponges, like some porosities in their skeleton, but also they have these septa, radial septa, which look like what you can find in corals, in cnidarians. And these archaeocetes appeared massively and were very dominant, let's say, only during the Lower Cambrian up to the Middle Cambrian. If we go a little bit further, so let's go to the end of the Lower Cambrian, something called Aldabanian, period called Aldabanian. We see the changing fauna, and the changing fauna in China marks what is really considered as the beginning of the Cambrian explosion, no? With a lot of forms, more than 100 different species, between brackets, 20 different phyla, and it's usually considered as one of the first modern ecosystems. And the Chengyang fauna here, it is represented by lots of arthropods, but also sponges and all kinds of worms and early chordates, so early animals which descendant would be the vertebrates somehow, and also some predators like Anomalocaris. If we go now to the Middle Cambrian, we have the burger shells fauna. This fauna was discovered at the beginning of the 20th century in 1909, I think, in Canada. And this fauna represents about 37 phylum, many of which are non-mineralized, but some of them are. So it means that the burger shell is really a good example of the Cambrian Sea, the Middle Cambrian Seas. And many phylum were already in existence there. I will not go into detail for this fauna. But most of the animals which were not mineralized had anyway an organic cuticle, probably a chitinous cuticle, chitinous exoskeleton to protect themselves. So if we sort of summarize what was the Cambrian explosions, it probably marks the apparition of novel organization plan for bodies, for animal bodies, for metazoan bodies. It's also characterized by the increase of diversity of the animals at all levels, from the species level, generic level, family level, order level, and so on. The Cambrian explosion marks the appearance of the modern marine ecosystems. It marks also some aborted experiment, like archaeocyates, which I was speaking about 10 minutes ago. Archaeocyates just, let's say, appeared in the lower Cambrian. They developed a lot, and then they disappeared in the Middle Cambrian, you know. And of course, the Cambrian explosion is really marked by the massive appearance of controlled biomineralization. So a lot of debate had occurred about the possible external cause of the Cambrian explosion and the possible possible causes of the appearance of skeletal tissues. Some causes are external, like tectonics, or like climate-related, cause related to the O2 in seawater, or the seawater chemistry. Some causes are more internal, like physiological causes, predation, genetic, and so on. So let's very briefly go to these different possibilities. Cause related to tectonics. At the end of the Precambrian, we have the fragmentation of the supercontinent called Rodinia. And this fragmentation really provided a lot of new epicontinental areas. So people say that the formation of a lot of epicontinental areas may be a driving force to promote the radiation of organisms. It is possible, but probably it's not enough. You have causes which are related to climate. The end of the Proterozoic is marked by the end of the snowball Earth, which may have had several consequences. One of them is the increase of sea level, meaning the formation of new ecological niches, but also the increase of fluxes of nutrients into the ocean. And of course, the end of the snowball might be due to a massive release of CO2 in the atmosphere, but this is not yet completely established. But it's possible that the end of the snowball may have, on the long term, the consequence of, let's say, promoting, not directly but indirectly, the formation of, or let's say, the occurrence of radiations. So if you look here on this plot, you see that the different metazoans here are listed, and you can see that they may be the appearance of this, let's say, different clays may be related to climatic causes. Here you have the different glaciation, Nantuo, Gastier, and so the post glaciation may be favorable of to the formation of or to the radiations of metazoans. We have also causes which are related to the level of oxygen in the oceans. And this is already a very old hypothesis, which was submitted by Tao in 1970. You know that collagen has a lot of, when you look at the sequence of collagen, you have a lot of amino acids which are a bit transformed, like lysine, which becomes hydroxylysine, proline, which becomes hydroxyproline. Having this, for the transformation of these amino acids, chlorine to hydroxyproline, lysine to hydroxylysine, you need oxygen. And Tao said that there was a link between the emergence of collagen and the level of oxygen in the oceans. So it's possible that successive thresholds of oxygen level could favorize the formation of the Ajakara fauna, then of the small shelly fossils, and then on the Cambrian fauna. So there is a complete literature about that. And here again, level of oxygen in the seas may have provoked or the appearance of new forms of life. Well, some causes are related to the chemistry of oceans. It's well documented that in the Neoproterozoic, for example in Spitzberg, you had formation of hypercalcified bacteria. And it seems that Neoproterozoic oceans may have been supersaturated with respect to calcium carbonate. And this environment may have favored the formation of calcified skeleton. Another point is that the magnesium-calcium ratio may have been modified during the transition between the Precambrian and the Cambrian. And this decrease may have also be a trigger mechanisms for formation of skeletal tissues. But besides these causes, which are typically external causes, which are typically environmental or chemical causes, you have also some ecological causes. So it's true that at the Precambrian-Cambrian transition, predation appeared. And as I mentioned 10 minutes ago, some cludinid samples were found to have trace of predation, but it was also the case on trilobites, on arthropods, which were chewed by predators. And one of them was the Anomalocaris, which was the killer organisms of the Cambrian Sea. Genetic causes are also, of course, many often cited as a potential mechanism for making new construction plan and so on, for promoting the radiation of metazoans. And it's true that there have been a lot of studies showing that animals, metazoans, diverge and radiated, and this radiation or these different radiation were associated to the implementation or to the emergence of new genes like the Hops genes, which are genes that code for transcription factors, which control the expression of other genes. And I will not go into detail in this area, but there is really if you look from more simple organisms here, sponges, to more complex, let's say, vertebrates, you see that the number of oxygen increase probably by duplication, and these duplication events probably allow the complexification of the body plans. But about 25 years ago, me and some colleagues, we proposed also a scenario about formation of the first skeletal tissue in the Cambrian, and we call that the anti-calcification hypothesis, where you had supersaturated ocean with respect to calcium carbonate, and this supersaturated ocean had the consequence for the organism to make some inhibitors to avoid overgrowth of crystals of their tissue. And among the inhibitors that were required, you may have had these mucus substances that we find everywhere. Mucus substances are these complex secretory products made of polysaccharide, but also proteins, like mucins. And this mucus may have been used as anti-calcifier, so to let's say, avoid super crystallization of animals' tissue. And then at the Cambrian transition, the inhibitors would have been reused for another function, not for anti-calcifying, but to keep crystallization in check. So that was our hypothesis about the invention of the early skeletal tissues. So if we go from a Proterozoic ecosystem where you only have soft-bodied fauna to Cambrian modern ecosystems where you have also mineralized fauna, you can say that few things are required. So to go from the absence of controls of mineralization to a controlled mineralization, you need first a specialized organic matrix. And as I said before, these crystals, which are formed for making skeletons, they have a certain hierarchy. So these are two properties. So if we, okay. Oh, sorry. If we go now to the Paleozoic, so after the Cambrian, so Paleozoic until the Permian times, so from 542 million years ago to 250 million years ago, a lot of things happened. So during the Paleozoic time, at world scale, all the biomineralization made by Metazoans, all the, let's say, controlled biomineralization were dominated by reef systems, which developed in the calcitic ocean. You have, for example, these Paleozoic corals, tabulate corals, rugosans. You have also what is called stromatoporoids, which are sponge-like animals. And these animals really made huge reef systems in the Ordovician, Silurian, Devonian, Carbonifera, and Permian times. But all these reef systems went on extinction at the Permo-Trias crisis 250 million years ago. And during the Mesozoic times, new players emerged. And these new players were the planktonic organisms, which were important providers of biogenic calcium carbonate. And we have the case of coccolith algae here. I will not go into detail, but if you look at the Cretaceous period, you know the Cretaceous was defined in the 18th century, in 19th century in France, and it came from the chalk, from the craie, we call it craie in French, and that gave the Cretaceous, because all these sediments, these huge pile of sediments, like we see in France, but also in England, are made of chalk, and these chalk cliffs are just are an accumulation of coccoliths. So 90% of the clay is made of coccoliths. So you have a huge accumulation of coccoliths in this area, and they probably were very important contributors of calcium carbonate during the Cretaceous period, and also later, but especially during the Cretaceous. If you look here on this satellite map, you see we have Alaska here, with the Aleutian Island, I think, here. And then this bloom, you see here in green, this is a bloom of coccolithophore, a bloom of Emiliania Huxley. So you see that even today, the contribution of coccolithophore algae is enormous in the global production of calcium carbonate. So this has the size of all Alaska. Huh? And during the Mesozoic times, something which happened was that the magnesium-calcium ratio started to fluctuate much more than it was fluctuating during the Paleozoic times. And this is what this curve tells us, meaning that this rate fluctuated, and when magnesium was high in the ocean, of course, it prevented the formation of calcite and promoted the formation of aragonite. When the magnesium flux was low, it promoted the formation of calcite at the detriment of aragonite. So during the whole, let's say, starting from the Permian to the Jurassic Cretaceous time, you had these fluctuations, and this fluctuation really marked alternance between aragonitic seas and calcitic seas. And during the Mesozoic times also, while calcifying planktonic organisms emerged, reef systems continued to develop, especially in the Triassic, but also the Jurassic and the lower Cretaceous. And the reefs were dominated by scleractinian corals, which are the equivalent of the today's corals. Only in the upper Cretaceous, scleractinian corals were sort of pushed aside by other reef constructors, which were rudist, so these big bivalves, sometimes 1 meter high, which were making complete reef systems, like you can see here, and you can see the size of these animals, and they could really make complete reef at the detriment of scleractinian corals. And it was like that for a few tens of million years in the upper Cretaceous. So to summarize a little bit what I've said, in brief, biologically induced calcification, it appeared very early in the history of the Earth via stromatolites, which dominated the biominoizations from the Archean to the Proteozoic. And then biologically controlled mineralization started to emerge soon before the Precambrian-Cambrian transition, especially via the Cambrian Explosion. During the whole Paleozoic times, the biologically controlled mineralization were mostly benthic fauna in an elliptic domain, so in platform domains. And only in the Mesozoic time, in the Trias and Jurassic, Cretaceous and later, you had a sort of mini revolution because planktonic protists started to take over, or partly take over, the production of calcium carbonate in open oceanic domains. So I have finished with my second talk. Shall I go to the next one? Is it okay if we continue?
Frederick? from Alberto, one of the students. And he's asking, it is related to the previous section when you presented the production of calcium carbonate on a global scale. And Alberto is asking, how is it, what are the methods, or how is it possible to estimate the production of calcium carbonate?
Well, you know, this is a key question, of course, because this was made by, you know, estimated locally and trying to, let's say, extrapolate.
Yeah.
You know, for example, for some systems, reef systems, if you look today where the reefs are, you can really estimate quite accurately the amount of calcium carbonate produced by scleractinians, for example. So the but I must say I didn't speak about that. There is a big, probably a missing actor, which are the deep sea corals.
Yeah.
So on the non-reef corals, which are living really in deep at 500,000 meters below sea level, and this we don't. There is no estimation of this. Maybe it's even more than what I presented. So this estimation is only an estimation. We can, I think, it's refined every time, but yeah, it's just extrapolating what is found locally. And you know, the thing is even worse in the fact that in some areas it's very easy when you just walk on beaches. You see that in some areas a huge amount of dead shells of let's say mollusks, bivalves, gastropods, and so on. So locally you can have huge accumulation, while a little bit later you don't see anything. So you know it's not. Yeah, this is an estimation.
Yeah. So thanks, Frederick. Tomorrow we will speak about classification rate a bit during my lecture. So
okay.
It's really related to what you were saying today to some aspect that I will tackle tomorrow. So I suggest I don't know, maybe we need a 5 minutes break and then we move to the next section. What do you think, Frederick?
For me, it's okay. I can go on. So if the students want to have a break, for me it's okay. Otherwise,
So what you want to do, students? You want to go on or have a break, 5 minutes? Hello?
So do they answer?
I don't hear the answer because the microphone is off in the room.
We didn't hear it.
We asked if you want to do a 5 minutes break or if you like to move on.
Can we do a 5-minute break then?
5 minutes break?
Okay.
Yes.
Okay, thanks.
Okay.
Okay, so please have a seat. We shall start the next section. Okay.
Okay, can you see my screen?
Yes, very well.
So shall I start or shall I wait a bit more? Well, I don't see.
Mariana, can we start?
I don't see the room.
Yes, yes.
Yes, the room is okay, it's full. You can start.
Okay. So now I'm going to specifically focus on the mollusk, on their shell biomineralization. So mollusk is a very interesting phylum because it's, after arthropods, it's the second biggest phylum on Earth in terms of number of species. One estimates that it comprises about 120,000 living species, but probably more. Mollusks are composed of 8 different classes. So mollusks represent a phylum, and this phylum is divided in 8 different classes. The 2 first classes, Solenogasters and Codophoveates, are made by animals which look like worms, with little spines on their teguments, little needles of calcium carbonate on their tegument. So they are very discrete. We don't see them so often. The third class is made of polyplacophorans, representing about 900 species. Then monoplacophorans. It's a very tiny group represented by 15 living species. And then you have a bigger group, including cephalopods, like Nautilus, or like the octopus. Cephalopods are represented by 700 species. Gastropods, and this is the biggest mollusk class, more than 100,000 species. Scaphopods, also called tusk shells, only 400 species, and then bivalves, more than 1,200 species. So this is the whole group, and this whole group appeared early in life history at the early Cambrian, as I explained just before. In particular, the monoplacophorans and the bivalves were probably the first classes of mollusks to appear, as we can see in the sediments. Then gastropods and cephalopods appeared a bit later during the Cambrian. And finally, the scaphopods appeared later, I think in the Silurian times. And the other groups we don't know exactly when they appeared because they didn't leave so much traces in the sediments. So since the Cambrian, mollusks have a complete, continuous fossil record without any interruption, in spite of biological crises, like the one of the Permo-Triassic, 250 million years ago, or like the one of the end of the Cretaceous, 65 million years ago. But they have this continuous fossil record. It's a group which had great adaptability and a great evolutionary success. You can think of gastropods which are able to live in all kinds of environment, high altitude, in deserts, in caves. Really, they have colonized all kinds of environments, polar environments and so on. And this success is mostly due to the shell, to the formation of a shell, because the shell is really an efficient shelter against predation first, but also an efficient shelter against desiccation. So even in dry places, you have gastropods which are able to live. And the shell is really a multi-layered material. I just show here the example of a freshwater mussel that we can see here in the surrounding of Dijon. In lakes, it's very common. You can find it in lakes, and it's an unionoid bivalve. It's Uniopictorum. And if you make a cross-section on the shell, you see that the shell is composed of few layers. First, you have this very tiny organic layer here, which is called the perostracum. So if you remember from my earlier talk, that the perostracum is the organic layer which provides this space delineation, so that the shell is not made directly in contact with the environment, but it's protected, it's sheltered, or it's closed from the environment. Then below the perostracum, you have here an external layer, which is made of these long prisms. And then below, you have an internal layer made of nacre. And in that case, the external layer and the internal layer are all together aragonitic. But in many cases, you can have an external layer made of calcite and internal layer made of aragonite. But all this is calcium carbonate. So if we look at the minerals that form the shells, in general, we have calcite, aragonite, we may have traces of vaterite, and we may have also a little bit of amorphous calcium carbonate. So you've seen already this picture. The shell is made by the mantle, the calcifying mantle, which is this thin organ, which allows the deposition of mineral. And again, this mantle works, it's a polarized epithelium, which allow the translocation of the precursor ion and their extrusion into between the mantle and the shell. You have also the secretion of amorphous granules. You have the secretion of the skeletal matrix by this organ. You have also proton reabsorption, because if we look what happens from a chemical point of view, we have bicarbonate reacting with calcium cation, forming calcium carbonate plus a proton. So you must reabsorb this one to push the reaction on the right side. Again, here I will not discuss too much in this, which I showed half an hour ago. The shell is really an organomineral material, because if you dissolve a shell, of course, most of the shell is calcium carbonate, but there is a little proportion which is made of organics. And these organics represent from 0.05% to 1 or 2% of the shell. And this organic is not made by one constituent, but actually by several constituents, among which one finds proteins and peptides, polysaccharides, probably lipids, which are very poorly known, pigments as well, and metabolites. And from here to here, we have really a gradient of knowledge from very well studied proteins and peptides, less studied, and these are very poorly studied. So the mollusk shell matrix is supposed to display major roles in biomineralization, and one considers it as the molecular toolbox used for regulating the deposition of minerals. But if you look from a historical perspective, there was a drastic evolution of the concept of the skeletal matrix or shell matrix just in the last decades, and I'm just going to show that to you. And the manner we perceive the shell matrix and its putative functions has a lot to do with the techniques we use to analyze it. So there is really a link between the way to apprehend this matrix and the way we see it. So I was telling you at the beginning of my talk that the first biochemical analysis of the shell matrix, of the shell organic components, were in the 50s, and probably 54 marked the very, very early work on the nacre, mother of pearl, made by biochemical techniques, amino acid analysis. So these analyses prevailed in the 60s, 70s, and the 80s, and they still continue. But on that are superimposed molecular biology approach, let's say from 1987, the very first works, and then more recently the omics approach, the highest throughput screening of shell components. And we are going to see these different phases. So first, the mollusk shell matrix was apprehended via a typical biochemical approach, and this consisted mainly in amino acid analysis on bulk matrices or not. People use chromatography, people use gel electrophoresis, and in the base cases, one could obtain partial protein sequences, very small sequences. You know, the first sequences that were published in the early 90s, in 91, 92, were just little piece of sequence of shell proteins. Not very informative, but at that time it looked very, very novel, you know. And then, if you look again this biochemical approach, the analysis of that time allowed to establish some models in which some proteins acted as molds around crystals or acting as physical templates. And this was mainly the case of hydrophobic proteins. And some proteins were supposed to bind calcium ions because of their polyanionic properties. And this was the case for aspartic acid-rich proteins. So in the, let's say, end of 70s, early 80s, emerged some models, especially due to Steve Weiner, Liad Hadid. And in these models, you had, as I say, soluble acidic proteins, which were more or less in charge of the nucleation inhibition. So they could act in two different ways, something like the yin and yang of the biomineralization. But you have also chitin fibers, which provided structural support. And chitin, of course, just to open a parenthesis, chitin is an organic polymer. It's a polymer of sugar, of a monosaccharide. It's a polymer, it's a complex polymer of N-acetyl glucosamine. And chitin is used in many systems. So it's an important component in the system. So beside the soluble acidic protein and the chitin fibers, you also have these insoluble hydrophobic proteins. So at the beginning, people thought that they were acting as mold, but later on, the group of Liad Hadid and Steve Weiner showed that probably these proteins were secreted as forming a gel. And actually, the crystallization occurred not in a liquid system, but more in a gel. So that was a model which was proposed in 2001. Then, for mollusk, came the molecular biology approach, and things started to be a little bit more complicated because a few proteins were, let's say, identified in few different models. In short, molecular biology means raising probes for fishing out the correct gene in, for example, in a cDNA library, or making PCR and so on. So by using these different techniques, people could obtain full sequences. Sometimes they could make in silico work and to do functional studies. But from the few proteins that were identified in the late 90s and beginning of the year 2000s, many of these proteins did not really fit into the model that I presented before, and new functions were identified. Of course, molecular biology was sort of progress in comparison to biochemical approach, but still it was a one-per-one approach, so meaning that you fish out one gene from a cDNA library, for example, you try to see what it does, and then you go to the next gene, try to see what it does, and so on. So it's a really one protein per one protein approach. Under this molecular biology approach phase, full sequences were, let's say, published. Between 96, which is the date of the publication of the first full protein sequence, nacrein, and 2008, we had about 40 proteins, or 40 shell protein sequences which were published, which is quite a lot, but finally not so much. Huh? For some of them, some overexpression work was done, and so on. And for example, you had nacrein, which is this protein which comprises some carbonic anhydrase subdomains, which are interspaced by acidic domain. You had the case of lustrine, where you have all these modules rich in cysteine and proline, a GS loop, and a C-terminus, which is a little bit different. So lustrine is supposed to be like a spire, and to work as a sort of connector between nacre tablets, for example. And you had also mucoperline, which I found, which had some modules, mucin type modules, and different domains in the C-terminal part. And again, its function was very elusive. So, in connection to biochemistry, to molecular biology, then started to emerge let's say, high throughput screening techniques, what I call the omics techniques, meaning genomics, transcriptomics, proteomics. And these techniques allow obtaining the complete skeletal repertoire, skeletome, shellomes, so the complete assemblage of proteins that are supposed to be associated to the shell and to be involved in the biomineralization of the shells. And there, when these techniques started around the around 2005, 2006, several additional molecular functions were identified. Well, I just summarize here how these techniques work. On one side, if you have an animal which is calcifying actively, what you can do is to take this animal, to sacrifice it, to cut the living calcifying tissue, meaning the mantle. From that, you extract all the messenger RNA from that tissue, which is supposed to actively calcify, which are converted to DNA. And then all these DNAs are sequenced massively via next generation sequencing. And the result is what is called a transcriptome or a secretome, so where you get all the genes or all the transcripts that encode proteins from the mantle tissue. In parallel, you can use proteomics, where instead of using the living calcifying tissue, you leave the calcified tissue, meaning the shell. And the shell, you will extract the organic component, the proteins from the shell. And via a certain number of operations, you will do some mass spectrometric analysis of the peptides that constitute peptides from the proteins of the shell. And if you confront the transcriptome to the proteome, you will get finally the proteins associated to the shell. You will get the skeletal repertoire, the skeleton. Well, this has been done for a number of mollusk models. Very few cephalopods. We've published a couple of things on cephalopods. Some more things are being published now. Gastropods, scaphopod, nothing, and bivalves. So nowadays, about 25 to 30 genera are now covered by these kind of techniques. Of course, it brings a lot of information, but at the same time, keep in mind that mollusk, 120,000 species, living species. So what we see here is very little tip of the iceberg. Yeah? So most of the things are still unknown. No? So the omics technique contributed a lot to the renewal of the knowledge about the shell matrices. So first, we got a massive increase of the number of identified proteins. And the graph I showed here is just describe this. Yep. Meaning that in '96, we had one full sequence of shell proteins. In 2004, we had 19 sequences of shell proteins. In 2008, we had a little bit between 40 and 50 full sequences of shell proteins. But due to the omics approach, we have now more than 1,000 full sequences of shell proteins. Okay, so we have a sort of exponential increase of the number of identified shell proteins. So which means that the concept of skeletal matrix is a little bit blurred in comparison to before. As I said before, we had several new functions which were identified, and many of them, a majority of them, are novel and unknown functions. So the question is to know if skeletal shell proteins, they are all involved in mineralization. And this is probably not the case. Here, I just symbolize the mineralizing epithelium. Here, this is the shell, and you have the space between the mineralizing epithelium of the shell, which is probably a nanometric or micrometric space. So the secretome are constituted of the protein which go through the secretory pathway. Probably some proteins are not at all involved in mineralization. Some of them are involved, but some of them are occluded in the shell, so they are really sort of when the shell is being secreted, these proteins are really associated to the mineral phase. And probably some of these proteins are not occluded, maybe even excluded from the shell, probably by hydrophobic interactions and so on. But beside these secretome, you may have some membrana proteins which extracellular domains are cleaved, and these extracellular domains may be occluded or not occluded. And you may have also as well some cellular contaminants and even nuclear contaminants, which might be occluded and not occluded. So we have here something a little bit more complex than what we thought initially. The fact is that we have more than 1,000 proteins, several functional domains, several protein families, and among these proteins, we have the one which comprise low complexity domains, and I will tell a little bit more later. So the problem that we face now is about the concept, the outline of the skeletal matrix. As I said before, the outline of the skeletal matrix are blurred. How to relate protein sequences and function? How do all these proteins work together? So what we can do now at the present is first to compare between different calcifying repertoires, to try to sketch some macroevolutionary trends, and also to work on one aspect which I will come later in my talk, as another talk, about how these proteins, when they are occluded in the mineral phase, how do they fossilize? So I will take a first example. This is the example of nacre. Nacre in mollusk is a well-known microstructure. It's actually the most popular microstructure. And we don't know if nacre was invented once or if it was invented multiple times. So nacre is one of the several shell microstructures that one can find in mollusk shells. It's always in an internal position in the shell. You see here nucula, you see here Mytilus, the edible mussel. What is white is nacre. You see here freshwater mussel, Unio. And here you see the shiny lustrous part is nacre. You see here abalone, and what you see shiny here is nacre. Pearl oyster. Here, Neotrigonia also, and here also, well, we don't see it, but inside you have nacre in the Nautilus. So the nacre has a broad repartition in mollusk. It's always aragonitic. It's a laminar structure. It's of Cambrian origin. I just show here two broad types of nacre. You have the brick wall nacre that you can see in many bivalves, where you have these tablets which are making like a brick wall. You see in different models. Here it's represented where you see here the epithelium, the mantle epithelium and the nacre tablets. But you have also some nacre which are a little bit more columnar that you can see in gastropods, for example, where the tablets are a little bit more one above each other with a little shift, you know. So this is nacre. If you look the distribution of nacre in mollusks, it's present in the Monoplacophorans, these tiny little groups of 15 species. It's well present in bivalves, in different orders like Paleotaxodont, Theriomorph or bivalve, Paleotherodont, Anomalodesmata. It's present in the cephalopods, mostly in nautiloids, because most of the other cephalopods, either they have a reduced shell or they have lost completely their shell. Huh? Think of the octopus, which has no shell. And it's also present in many gastropods of one order, the vetigastropods, including the abalone. So by doing proteomics, we are able to show that finally, by taking three animals, one mussel, one pearl oyster, one freshwater mussel, we could see that some proteins are shared between the three groups, which are already quite distant from an evolutionary viewpoint. But some other proteins are very, they are more specific of the group, and they are not found in the other group, in the other two specimens, like these proteins that are found in the unionoids, in the unio, these which are found in the pteriomorph and in the pearl oyster, and these which are found in Mytilus are really unique to these animals. So we have a mix between shell proteins and lineage specific. But if you look now also between, let's say, pearl oyster and abalone, here you see that they have in common only one protein. So which means, or which suggests, but only suggests, I don't want to go too far in the interpretation. It means, it may signify, it may means that nacre was invented a couple of times, was invented in gastropods separately from bivalves. So because here we have very different nacre assemblage. So we cannot exclude neither that there is a fast evolution of nacre protein and that there was a sort of drift from a common nacre ancestor. What is interesting to observe is that here, with these two examples, the bivalve examples and the bivalve gastropod example, we have a different protein assemblage can build similar microstructure. So it means that the system has a certain plasticity, and we still don't understand what is the driving force which constrains the system to produce nacre. If we use different proteins for making the same the same type of microstructure, so what drives the system? What constrains the system to produce nacre? There is a second example. This is the one of prism and nacre. And this was an example we've treated about a decade ago when we were in a consortium focused on improving the quality of pearls in French Polynesia. So you know, French Polynesia is the main producing area for black pearl, the Polynesian pearl, and which is made by the Pinctada margaritifera, the black lip pearl oyster. So you see why black lip? Because you see that here it's really black. Huh? There is a lot of pigments in this area, or in the nacreous area, but also in the outer layer, which is prismatic. And if you look at here in cross section, you see that you have really two layers which are very, very different. The prismatic layer is made of calcite here, and the nacreous layer here, that you see bigger here, is made of these tiny tablets of aragonite. These tablets, they have about 0.5 micron in thickness, and they have 3 microns in length, while the prisms are really looking like big crystals. So we had the question that, are the prisms and the nacre made from the same shell matrix repertoire? And this question looks rather simple or rather naive, but it was actually running for decades. I just refer here to a few papers. The paper of Philip Hare in Science in 1963, where he made amino acid analysis from the proteins from aragonite and calcite in the shell of Mytilus californianus. So Mytilus, the edible mussel from California, has the same kind of shell structure, meaning prism, calcitic prism, and aragonitic nacre. And he showed that the bulk amino acid composition of this animal was different depending whether you analyze prism or whether you analyze nacre. Then in 1967, Charles Grégoire, a guy from Belgium, also showed that organic matrix associated to shells, prismatic and nacreous shells, they had different amino acid composition. And Steve Weiner, in a paper published in Biochemistry in '83, he showed that Mytilus californianus, that's the same animal, had so he could, let's say, separate the shells of the shell layer of this animal, and then look at them from a biochemical point of view, and he saw differences. So this question was running for quite a while. And actually, by doing proteomics associated to transcriptome, we found a complete listing of proteins of a prismatic layer, so the external layer here, that is here on a gel, on the nacreous layer here, so this internal part. And I will not, of course, go into detail into this protein list, but what I can tell you is that these proteins are quite different, really quite different. So here with this work, we identified several markers of shell calcification, 48 present proteins, among which 40 were new, 28 were complete, 32 nacre proteins, and so on. And we showed that these proteins these protein repertoires were very different. So I just summarize here what we found in terms of protein domains and function. And if you look at the overlap between the prism repertoire and nacre repertoire, only few proteins are shared with the two layers, which is quite fascinating. So for the protein domains, we found a lot of proteins which have what we call low complexity domains. So these are domains which are dominated by one or two amino acid residues, like glycine-rich domains, alanine-rich domains, aspartic acid-rich domains, and so on. So these proteins are really something that you see a lot in biomineralized tissue, not only for mollusks, but also for other groups. So you had a lot of these proteins that contain low complexity domains. You had proteins with extracellular matrix like signature. You had enzymes. You had a new, a completely unsuspected class of protein or family of proteins, proteins containing protease inhibitors domain. And you had also proteins with EFN, which are supposed to be calcium binding proteins. So this is what we found. With a collaboration with German colleagues, with Daniel Jackson from Göttingen, Daniel was working on the Limnea stagnalis. So Limnea stagnalis is a freshwater gastropod. It's a pulmonate gastropod, so sort of modern gastropod, so to speak. It belongs to a superfamily that emerged 140 million years ago, something like that. It's not at all nacreous. Its structure, shell structure, is so-called cross-lamellar. And so Daniel worked on this, and we did in Dijon the Proteomics. And what is nice to see, so that's what Daniel and his colleague showed, that Limnea stagnalis, if you see on this circular graph, you see the similarity. Each little stripe means one protein. So here you have the complete repertoire of Limnea stagnalis. And some of the proteins, they have similarities, some kinds of similarities at different levels, global similarity, top quartile global similarity, third quartile, and so on and so on. So some of these proteins, they have similarities with the proteins of other calcified systems. And you see here that Limnea stagnalis has a lot of similarities with Cepae nemoralis, which is the garden snail. They are both, let's say, modern gastropods, but also it has some similarities with pearl oyster, with Mytilus coruscus, the edible mussel, Crassostrea gigas, so the oyster, edible oyster, Haliotis, very few with Haliotis ligata, and so on. Almost no similarity with the echinoderm, Strongylocentrotus purpuratus, but still some similarities with a cnidarian for a couple of proteins, and also with Lotia gigantea. So here we have something which looks like a very mosaic picture of similarities. So the skeletal proteins, or the shell proteins, if we want to sort of summarize what we know nowadays, let's say per shell, depending on the way we look at them, if we look at them via proteomics, it goes from 2 dozens of shell per shell to more than 100. Most of them are modular, they are multifunctional. Some of them are a little bit chimeric proteins. So their functions are deduced from sequence analysis and comparison to known proteins which have clearly identified functions. So some of the domains identified have similarities to those found in vertebrates, particularly the proteins which have extracellular matrix domains. There have been functional assays to identify the function of these proteins, but in a very few proteins. And if we want to summarize what we know about these shell proteins, we have the shell protein group constituted of proteins with domains of clearly identified functions. And these domains are the following: enzymatic domain, sugar binding, protease inhibitor domain, calcium binding, immunity related, or extracellular matrix-like domain. And we have a huge set of proteins of unknown or putative functions because they don't have any homologues in other systems. And most of the proteins which exhibit low complexity domains or repetitive low complexity domains are in that group, and also some other proteins which do not have low complexity domains. So this part is really like an unknown story. So to summarize, and I think, oh, sorry, whoop. What did I do? To summarize my talk, so skeletal matrix proteins or shell matrix proteins, there's a huge diversity, which probably means a high plasticity of the system. We have a modular architecture of the sequence of these proteins. We have the abundance of low complexity domains and repetitive low complexity domains. And this probably means that we have a very complex macroevolutionary history of these proteins. Some of the proteins were probably recruited very early, probably during the Cambrian or just before. And these proteins, these genes, which were recruited early, had a lot of secondary evolutions in the different lineages. And some other proteins were probably recruited very recently, and these proteins or these genes are more lineage dependent, and they also have this capacity of a fast evolution. And I would say, this is more or less what people think now, that the proteins which have low complexity domains have been recruited rather recently and are more lineage specific. Which means that these proteins, the proteins we deal with, these shell proteins, they do not carry really a phylogenetic signal at high taxonomic level. So we cannot use them for doing phylogeny, for example. So this is the end of my third talk. I can, if you want, I can go directly to the. Is it okay with you?
Yes, for me it's okay. I don't know the students.
Okay, so I can, I have a small presentation here.
I can go.
So maybe if you have question, because I'm speaking, but I hope that people can, that the students are surviving.
So Mariana, what do you like to do? Do 5 minutes, or you want to go on? I didn't understand. It's always the same question.
Go straight to the next one.
We go straight to the next one?
Yeah.
Yes.
I've got this one which is rather small, and then if people are not dead, I've got a fifth presentation which can be long, but I can make it short. Okay, so let's go to this one maybe. Huh?
Okay, thanks.
So in the previous talk, I was speaking about shell matrix or shell protein diagenesis. What is diagenesis? Diagenesis is all the processes that occur when you bury something in the ground, in the soil. So you have fossilization, or you have things get fossilized. So diagenesis is the transformation of a mineral phase or an object with time. So we've been involved in this kind of research too. Here, as a preamble, the calcifying matrix in metazoans, like Cnidarian, Bryozoan, Brachiopods, Mollusks, Echinoderms. As I said before, the organics is only a little part of the matrix, of the full skeleton, from 0.05 to 1 or 2% of the skeletal tissue. And most of the shell of the skeletal tissues are calcium carbonate, plus minor element, traces elements, and so on. Magnesium, strontium, and so on. Sulfur, and so on. So, again, going back to the graph I was showing, to the drawing I was showing 1 hour ago, 2 hours ago, in the neritic environment, biogenic carbonates are about 2.5 billion tons a year produced. This production is benthic, made of aragonite, calcite, or magnesium calcite. But if you look, so if you bury all this, that becomes the fossil record, it means that you also bury the matrix. So with the matrix representing, with the matrix representing 0.05 to 1 or 2% of the weight of the skeletal tissue, it means that per year you bury 1.2 million tons a year of calcifying matrix, from 1.2 million tons a year to 50 million tons a year in neritic environment, which is quite a lot. So when the animals die and when they are progressively integrated into the fossil record, you have here a very important source of biomolecules in the fossil record. But how do all these organic molecules fossilize? This is a big question. So this big question was started to be tackled by Abelson in 1954, who probably was the first in the world to make amino acid analysis of fossil shells. And he published that in the Carnegie Institution of Washington yearbook in a milestone paper, some aspects of biochemistry also. And Abelson is probably the founding, can be described as the founding father of the paleobiochemistry or even the molecular paleontology. And since the work of Abelson, there had been, especially in the mid-50s, in the 60s, 70s, two types of approaches to understand how organic matrix can fossilize, or how can it survive a long time, long geological times. One was to rework on fossils, to do some matrix extraction and analysis. One was, the second approach was to do some artificial diagenesis experiments to heat shells, for example, to see how to look at the way proteins can degrade. And from a historical perspective, we can say that from the work of Abelson until the early 90s, this was considered as a golden age of fossil protein analysis, especially protein analysis in shells. And there was this book, The Biogeochemistry of Amino Acids, which was published in 1980, which at that time summarized very well what was known about the degradation of shell proteins and the potential of shell proteins to overcome the long geological time. But somehow this research was completely, let's say, swept away by one discovery, and discovery was a PCR, polymerase chain reaction, which gave the possibility to look at fossil DNA. And of course, looking at fossil DNA was somehow looked easier and completely, let's say, shadowed the work on the fossil proteins. It completely overshadowed the work on fossil proteins. So to make things very simple, I just show here the different reactions that occur for the degradation pathway of skeletal matrix protein. First, shell proteins can be hydrolyzed. Partial hydrolysis will generate peptides, which themselves can be cut in free amino acids. But amino acids can be racemized, so going from the Lavoisier conformation to the Dextrogier conformation until reaching a certain equilibrium. You have also defunctionalization of the amino acids. Amino acids can be submitted to decarboxylation, desamination, giving birth to amine, organic acid, hydrocarbons. And you can have also condensation. So amino acids can condense with polysaccharide and make via what we call the Maillard reaction. You know, this reaction that occurs when you cook meat, for example, when you make this brownish stuff. This brownish stuff looks like humic acid. So when you have all these processes working together, at the end it's very difficult to see what has happened finally. So the hydrolysis of skeletal matrix protein depends a lot of different parameters. First, per se, it depends on the stability of the peptidic bond. One has to know that, for example, a peptidic bond between one alanine and one glycine is very, very stable. But a peptidic bond between two aspartic acid residues, it's much more labile. So intrinsically, you have really a complete range of stabilities of the peptidic bonds in a peptide chain. It also depends on the structure of the protein, if the protein is completely folded or uncoiled. It also depends a lot on the post-transactional modification, like glycosylation, phosphorylation. It also depends on the localization of the protein in the biomineral. Here we have the very simple drawing symbolizing nature tablets. So if you have intercrystalline proteins, so between nature tablets or intracrystalline proteins, they will not behave the same way. Intercrystalline will probably degrade first because water, they can be accessed by water molecules, so they can be easily degraded. And then if water makes start to have access to that, it means that bacteria can have access. So you have a complete pathway of degradation of these intercrystalline proteins. But for intracrystalline, that's another story because they are somehow protected by the surrounding mineral phase. They are not accessed easily by bacteria or by fungi, So they can probably stay longer in the mineral. It also, of course, depends on the water, if the water is interstitial or if the water is really linked to the structure. And of course, it depends a lot on the polymorph of calcium carbonate, calcite versus aragonite. We know that the calcite is the most stable form of calcium carbonate. Aragonite, is a metastable form of calcium carbonate. And so aragonite on the long term has a tendency to degrade, to transform into calcite or to recrystallize. But here on that picture, I was showing some nacre tablets that I took from a nuclear species of Lutetia, so 45 million year old specimen from the Lutetia of Paris, I think. So if we look here, the aragonite is really seems to be very well preserved. So, and I will just show here a few examples we have treated in the past or even recently about degradation of these shell proteins on their stability along geological time. So we did some experiments about the thermal stability of nacre proteins. So we took nacre that we bleached in a certain way. This nacre, we heated the nacre powder at 100 degrees during 10 days. And every day we were sampling an aliquot of this powder. And then from each aliquot, we were extracting the matrix, we were quantifying the matrix, we were doing ELISA, we are doing proteomics. And for example, by doing ELISA, by doing immunological tests on the intracrystalline matrix, we saw that from T0 to T10, there was a decrease of the signal, of the immunological signal given by antibodies. But this decrease was not very effective. I mean, there was a decrease, but still the signal was quite good after 10 days. And here I used two different antibodies. And here there was only a loss of 20% of the reactivity of the sample after 10 days of heating at 100 degrees. Doing proteomics give a little bit different picture. Some proteins at T0, T5, or T10, so T0, 120 hours, 240 hours, could be completely retrieved. So they just overcome the heating and they did not degrade. And some of them, like Perlin and so on, we did not find them after 5 days of treatment or even after 10 days of treatment, you know. So it means that we have here probably a differential degradation of some of the proteins. Some of the proteins have a certain persistence. Some of them looked apparently degraded. And also, if you looked at these red numbers, these are the numbers that these are the number of peptides that identify the protein. For example, here at T0, nacrine A1 is, let's say, recognized by 13 different peptides. But after 5 days of heating, only 7 peptides recognize nacrine A1. And after 10 days, only 4 peptides recognize nacrine A1. So globally, we see a decrease of the number of peptides that allow the recognition of the protein. So here in our system, where we just heat a shell powder, we see the disappearance of some proteins, but we see that some proteins are persisting, but they are recognized by a decreasing number of peptides. A second example, this is the example of subfossil Tridachna from French Polynesia. And this is a collaboration that we had. French Polynesia, you know, it's a big set of 5 different archipelagos. and Gambier, Australia Island, Society Island with Tahiti, Tuamotu, and the Marquise. And most of them, you have high island and low island. The low island are the ones which are made of lagoon and an atoll. So in that case, we had specimens from the Mangareva, from this atoll, which is a reef flat, rich in percolating water, which is absolutely not favorable for good preservation of aragonite. So our question was to know if we still had a biochemical signal staying in these shells. So this is a young shell of today, which was collected, and this is a subfossil shell, so it's completely covered with crust. And this shell was dated about 3,320 years old. And we made a control of the mineral phase with thin section, SEM, cathodoluminescence, epiphorescence, and so on. Oh, sorry. We could see that the outer layer was heavily attacked by andolites, or let's say organisms, probably fungi, but probably also bacteria, and so which could remake holes. But the lower part of, let's say, the internal part of the shell layer was much more clean. So this animal has is fully aragonitic. It's a cross-lamellar and complex cross-lamellar microstructure. We could see via FTIR that it was still aragonitic with this double peak of aragonite. Again, important alteration in the outermost or innermost layer of the shell. And then we extracted the organic components from the shell, especially in the intermediate shell layer. And we analyzed that on gel, and this is what we obtained. And we also looked at the proteomics, and we saw that we have here the fresh sample, which is recognized by, which indicates that it has a lot of protein, which is normal. But the subfossil samples, like two of them here, they also contain still some protein in spite of their preservation state, which is not so good. And we could even really look at some proteins like this one on the modern sample, which this protein, which is unnamed because we don't know what it does. This protein is covered by at 25% of its sequence. And here, the same protein, but from the fossil, is covered at 15% by peptides. Here, again, another protein, another one, covered at 30% on the fresh sample, and here covered only at 11% of the fossil samples. And here, we have a protein which is very well covered on the fresh sample, 38% of coverage. But here on the fossil sample, we only have a very low coverage, 4%. So which suggests that we have here, we can recognize some proteins, but because we don't see the other the peptides, some peptides that are here, for example, are not seen here, it may mean that the protein is getting degraded or has some alteration of its sequence. So that with this example, we can really, what is nice is that we can really sort of follow the degradation of proteins by proteomics. Now I would like to speak about the third example. And the third example is very nice. This is something we had to treat together with my colleague Beatrice De Marchi in Torino, in Turin. And that was in the frame of a co-tutel thesis, the thesis of Jeroen Sakala-Oskate, who she defended last January 2021. And she's now postdoc. And actually, we wanted to see the main scope of the thesis was to see if proteomic techniques can be applied to archaeological samples. And there was an example of these archaeological samples, which is represented here. This is what we call the double button pearls, in German Doppelknöpfe, where you have this tiny little piece of shells, which were really delicately made by the prehistoric populations a few thousand years ago. And they were using this, they were making these sort of buttons with a little hinge in the middle, which was why a hinge? It was for inserting these pieces in the leather. So for making ornaments, in their clothes. So it was a complex process of fabrication, you know. The thing is that having this piece of material, you absolutely don't know where it comes from. Meaning you don't know the biological origin of these Doppelknöpfe. And so one of the goals, one of the scopes of the thesis was to see whether by using proteomic techniques we could really trace or track back the biological origin of these little buttons. And here I just mentioned the fact that what was done a few thousand years ago with these little buttons is a little bit like what you find in England. When you go to England, you have these pearlies, you know, these people who cover their clothes with naked buttons, you know. So somehow the tradition is getting propagated from the old time to until now. So there were three archaeological sites, sorry. One was Havno in Denmark, so close to the sea actually. One was Hornstadt in Switzerland, and the third one was in Pestera Ungureasca. Sorry for my poor accent. Pestera Ungureasca in Romania. And these three sites are approximately contemporary now from each other. So this one is from 4,420 to 590 before Christus. This one is a bit more or less the same, and this one is the late Neolithic, beginning of Bronze Age. Okay, but all of them have these little piece of little buttons, little Doppelknöpfe in the. So the idea was that there were long distance trading to bring shells from, especially from the Mediterranean Sea, to propagate these raw material across Europe. You know, it was more or less the idea that the idea that people had about the use of this material. So we analyzed the buttons, these little Doppelknöpfe by SEM. So you see that they are really like nacre, typical brick wall nacre, and typical brick wall nacre means probably bivalve origin, but we cannot go much further because in bivalves you have different groups in bivalves which are able to make this type of nacre. Then we looked, some Doppelknöpfe had some little remains on the top, which were kept, not abraded, which fortunately, and probably used, maybe also it was a part of the ornamentation. And we had a prism, and these prisms were aragonitic. So this prism suggests that this group, Paleoheterodonts, may have been used, but it was not a very strong evidence. But it was coupled with stable isotope geochemistry of delta O18 and delta carbon 13, and which gave some very negative signal, which suggest a freshwater or estuarine origin. But the main argument was that when we micro-extracted the protein remains from these Doppelknöpfe, it's not a lot of material, I can tell you, we identified 3 proteins like HIC74, HIC52, silk mapping, which are really proteins found in unionoid bivalves. And unionoid bivalves are freshwater mussels. So we have here these 3 proteins with some of their characteristics. So we without any ambiguity, we demonstrated that all these Doppelknöpfe were made from freshwater mussel nacre, from Unionoidae. So actually, these proteins were not at all marine. They were just local freshwater shell material. So the tribes, the prehistoric tribes of these 3 locations just exploited local freshwater resources for making button pearls. So no use of marine nacreous shells. The ornaments at that time were probably a sign of prestige, but the concept was not associated to the rarity of the resource. But what made the button pearl rare was the effort made to fabricate them. So the resource was probably abundant, but what was the true bottleneck was really to make them properly. So we cannot advocate for a long exchange trading of raw material, but there was rather a propagation of the know-how to craft double button pearls. So with this example, we have shown that proteomics can be very powerfully used for archaeological questions. So to summarize a little bit what I'm saying now, you have a persistence of some shell proteins after long diagenesis. So it means that there is good hope that we can find fossil proteins in very old fossils. But these proteins of the shell matrix, they have a differential degradation patterns. So some of them may be resistant, some of them will be degraded very easily. If you look at diagenesis from the proteomic viewpoint, we see that some proteins disappear early, some of them persist, but they are identified via decrease of the number of identified peptides. So the main message is that now it's time to revisit diagenesis process by proteomics. We can track also diagenetic behavior of even one given protein within a shell. And we can really revisit archaeological question and also go further, I mean, go to all the samples, not only archaeological. Well, that's it for my 4th presentation. So if you want, I have another one, which is a little bit different from the others. I can sort of pack everything in half an hour. What do you say?
In half an hour, we are still in, if you can.
So what
said in half an hour, we are still in the time. So
yeah, okay. So what shall I do? Shall I?
Is the biotech application?
Yeah, it is more about the different use of biominerals. It's also sort of very general talk, but if people are interested, I don't know.
It depends on the student. For me, it's okay. Let me ask to the class. It's okay.
Yes, it's okay.
Okay. Let's go to the last layer of biomin science. So can you see the screen?
Yes.
Okay. Okay. So in this last half hour, I will just speak briefly about several applications that we can do with biominerals. So again, I presented this drawing first to show that the biomineralization as a discipline is at the crossing between many different disciplines, from very fundamental disciplines to more applied. And now we are going to look more to applied aspects. First, we will start with health. So when I say applied, I mean that the study of biomineralization can really have an impact on the how we can cure people, how we can improve the knowledge about about the biomineral system. Well, first, very easy to understand the application, the dental surgery and implantology. So we know that the implants represent a huge market, 3.5 billion euros. Well, I don't know. Yeah, I think at the world scale probably. And this market has a huge high added value. And of course, knowing the process of tooth biomineralization, how teeth grow, is of course has a lot of incidence on the way we can treat problems with teeth and make implants and improve the implants. The same is for bone surgery. Huh? We live all in Europe. In Europe, the population is aging. A lot of population, especially women, have problems of osteoporosis. And this is one point, but we have also problems with the hip, knee implant, and prosthesis. I just gave the numbers in France per year. Knee implants are about 30,000 prostheses, hip about 100,000. In the USA, it's you multiply everything by 3. And of course, this field is dependent on the progress we made on understanding how implants work and how biomineralization work. I will not go further. Just about the bone surgery, a niche market which is made by coral implants, especially from the Porites species. We know that in the 80s, it was found that coral could be used as implants for bone reconstruction. Nowadays, corals are decontaminated and they are completely converted to calcium phosphate by a thermic conversion. And they make little implants from them, little cylinders, which have a good porosity, which facilitate the migration of osteoblasts. They have relatively good mechanical properties, but they are brittle, of course. But what is interesting is that they have a good resorption capacity. So these implants are, again, a niche market, but they exist. So it has to be noted. Well, in nephrology and urology, we have the problems of kidney stones. At the beginning of my talk, I was speaking about organic biominerals like anhydrous uric acid kidney stones and also oxalate. And of course, it's a big problem, big health issue. 80% of these kidney stones are calcium-based, with calcium oxalate, vidylit, calcium phosphate, carbonate, apatite, brushite, and some of them are non-calcium-based, so in 20% of the case, and mostly uric acid, 10%, and it's usually when you have problems with gout. But struvite also, magnesium phosphate, or cysteine, xanthine for 1%. So, few representation of coralliform kidney stones, calcium oxalates. these it's interesting to see that there are really some inhibitors of the formation of this biomineralization, like citric acid, magnesium, oligoelements, or glycosaminoglycans, lithocalcine, uropontine, pyrophosphates. So there is a complete research devoted to the possibility to avoid the formation of these kidney stones. Uric acid kidney stones are also something problematic, which can be cured by also different inhibitors or different treatments. Beside the kidney stones, you have the pancreatic stones. Also, a lot of people are submitted to these problems. These pancreatic stones can be also avoided owing to some inhibitors like lithostatins. So there. In pharmacology, there is a true need to discover natural bioactive substances that can be beneficial for health, which are extracted from biominerals. And we have, again, the well-known example of nacre. Nacre is used in bone surgery, was used in bone surgery, and it was shown that nacre had osteoinductive and osteogenic properties. But now it's used in cosmetology, in dermatology, because nacre extracts have good, they are quite efficient for dermal applications. Again, pharmacology, food supplement. You can have food supplements made from male, from red algae, nacre powder, cocolite extracts, organic silica, which is in reality diatoms. So if you look on the internet, on the websites, you see a lot of products that are sold, maybe not all very efficient. It's more like poudre de perlimpinpin, as we say in French, meaning that, but there is a true market for this kind of products. Let's go to biomaterials. Well, biomineralization can be used as construction materials. Of course, limestone, especially when these limestones are made from coral deposits and so on. You have, I was mentioning the chalk, and the chalk is also used for making lime. Other materials, diatomite. Diatomite is the rock made from the accumulation of diatoms, algae. So diatomites are very specific rocks, usually very light. So they are used for filters, for example, for wines. They are used for abrasives, for depolluting, as a mineral filler for paintings. And we have natural deposits, for example, in California. The mine of Lompoc is the biggest in the world. But Turkey, China, and are also big producers of diatomites. In total, the world production is about 1.8 million tons a year, and 50% comes from the USA. And diatomites were made in the past, especially in the Cenozoic time, in coastal marine deposits or lacustrine deposits in relation to volcanisms. So diatomites is really a rock made of biominerals, only of biominerals, which can be used for several applications. Now let's go to more specific things. Biomimetic materials, bio-inspired materials. As we know, most of the processes that are used in the industry for making materials are very costly. For example, the sintering of ceramics is made at high temperature, so it costs a lot of energy. And also, if not costly in terms of temperature, it can be polluting for the polymer chemistry. And of course, in the last decade, last 20 years, people wanted to promote the development of green chemistry, which was based on the synthesis, process made at room temperature with a minimum of energy. And of course, if you look at nature, nature fabricates biominerals under these conditions. Room temperature, minimum of energy. So observing nature and try to mimic what nature does is a sort of a bottom-up approach. So there are a few examples like that, like nanocages, nanotubes that are made of silica or other materials. And they are completely inspired from the diatom frustule, from the diatom test. And the idea is to use this nanocage for delivering active substance in the body. You know? So here you have pictures where you see A and B are, I think, natural nanocage, and this is a titanium oxide replica of a nanocage. Again, in the biomimetic material, you have, for example, the siliceous sponge spicules, and some of them behave like optical fibers. I've seen this spicule, which is in the property of Professor Werner Müller in Mainz. And when you apply the light here on that side, you will see that the light is completely propagating through the spicule. So the spicule really behave like optical fiber. So we can regain some knowledge about looking at these spicules and trying to mimic them. Materials in tablets that mimic nacre and its microstructure. As you know, nacre, we spoke a little bit about nacre, but nacre is a tremendous material because its resistance to fracture is about 1,000 times higher than that of chemical aragonite. And of course, calcium carbonate is a very cheap material. So the idea would be to sort of be able to mimic the structure of nacre with these very flat tables which are grouped and which do not allow propagation of fractures very easily. So some attempts have been done in the last years where they found a process that mimic nacre and its microstructure. And they made, of course, a nacre and aluminum oxide, which is maybe not the best material especially for the body, but they were able to really make something like nacre tablets, you know, of course less solid, but people are improving their process now. Well, about the material of the future, we can think about the organomineral hybrid material for nanoelectronics or semiconductors. And for that, people use the phage display peptide libraries. So I will not explain very lengthily, but this is a very beautiful technique where you use combinatorial libraries of phage particles that contain peptides in their coating. And these peptides, so these libraries are put in contact to a mineral surface, and only the peptides which have an affinity, a certain affinity with the surface, will bind to the mineral surface, and the other one will be washed away. So all the unbound phage will be washed, and when you repeat this cycle, you end up with selecting peptides that are very tightly bound to the mineral surface. Then if you sequence the phage, you get the sequence of the peptide, and then you can really design specific peptides which have a very strong affinity to the surface. So this technique has been used a lot of time for making these multi-layers material on like semiconductors and so on. About environment, biomineralization are very useful for different reasons. First, for biofouling. You know, biofouling is colonization of surface by living organisms that are able to make crust and then degrade environment. So the threat are really endolytic microorganisms. So they can be this threat can be removed by either by using calcifying bacteria to remineralize construction material, degraded pollution, but it can also be used, fought against by using natural antibacterial substances. About the anti-fouling, we have also the problem with boat hulls. So you know that super tankers, gas carriers, they if they don't have any anti-fouling on the hull, there is, when they go from one point to another, there is an increase up to 40% of fuel consumption. And I show the example here of a hull of a boat, where you can have until more than 25,000 marine species which are able to colonize a submerged surface. 1 or 2 millimeter of encrusting algae is about 15% of loss of speed, and 1 square meter of boat hull, which is colonized, can welcome up to 180 kilograms of organisms. So there is a complete sequence of colonization by bacterial biofilm, Foraminifera, algae, barnacle, mollusks, calcifying worms, and substances extracted from biominerals. Some of them have these anti-fouling capabilities. So it may be possible that later some process will be found to extract or get some, at large extent, some anti-fouling substances from biominerals to be used as natural anti-fouling. Yeah, I didn't say that chemical anti-fouling are very, very dangerous and very polluting, like TBT, tributyltin. So again, environment. Yeah, biominerals are used as ecological monitoring. Biomineralization are the ecological sentinels of the environment. Biomineralization are the archives for environmental parameters, temperature, salinity, and pollutions, of course, heavy metals, organic pollutants, pesticides. Also, CO2 storage. You know, we are living in a world where the increase of CO2 is a reality in the atmosphere, but also the acidification of the ocean, which is correlated to the increase of CO2. Because now the ocean, in the pre-industrial period, the pH of ocean was about 8.2. Today it's more about 8.1. And probably in 100 years or less, it will be 7.8. So using biominerals to store calcium CO2 is maybe a possibility. Of course, for the moment, it's kind of unrealistic. But trapping CO2 with calcifying organisms like calcifying bacteria may be a way to really decrease the amount of CO2 in the atmosphere and in the ocean. Water depollution. I've seen that with my eyes. It's very impressive. So biominerals, when they are reduced in powder, they have interesting charge surface properties, which is due to the presence of organic polyanionic macromolecules. And with this, you can really depollute waters, especially waters which are full of clay minerals that are in suspension. So during flood, for example, you can really depollute waters just by using shell powder. And the other thing is that they are able to trap metallic ions, and they can also alkalinize acidic water. I was referring to the soil amendment, soil poor in calcium, magnesium, acidic soils. So this is a very simple application of biomineralizations. Aquaculture and permaculture, that will be, I think, my last example. Zootechnics, aquaculture, shellfish farming, We have a lot of pathology which are affecting the shells of edible species. In the 80s, you had the chambering of oyster shells because of TBT, tributyltin. Now it's over, but it was a real problem. Scallop shells and also Minia clam, which were affected by brown ring disease that affected the shell. So using these, using the knowledge, using that we get on biomineralization, we can sort of overcome these type of problems. And I will finish with the pearl culture. As you probably know, pearl culture is a huge industry in many countries. I just refer it to French Polynesia, with again these 5 archipelagos: Austral, Gambier, Société Island, Tuamotu, Marquise. The main centers of production of pearls are the Tuamotus and the Gambier Islands. And it's a huge activity, like 800 pearl farms. 90% of the black pearls in the world are made in Polynesia. And 58% of the exportation of Polynesia is about the pearls, which generate about $120 million per year. And the situation was not so good about 20 years ago, because the price of the pearl went down, or the price of the average price of pearl per gram produced was really dropping down. And it was a true crisis because of the loss of quality, decrease of the quality of the pearls. So that's why there was this group of researchers which was set up about 15 years ago with different partners, and I will not show that. Well, this is pearl culture from Polynesia. Well, I just show here how to fabricate a pearl. You know, pearl is a complex process which has a lot of empirism, but also some very strictly controlled things. First, you have to get a donor oyster. Donor oyster will be used for, will be sacrificed, and one can take a piece of the mantle, a ribbon of the mantle, which is cut again in little squares, like called graft, and the nucleus on the graft are inserted together in a recipient oyster, and the recipient oyster will make, because of the graft which is here, it will make months after months, weeks after weeks, a pearl sac after 45 days, and from these 45 to 60 days, the mineralization will start to occur around the nucleus, and then after 18 months, so 1.5 years, you get a full pearl, which is thick enough, which has a thickness more than 0.8 millimeters of nacre, so it can be commercialized. And of course, we did some work. When I say we, all the consortium did some work to exactly understand the process of pearl making, and also what kind of graft we could use to improve the color quality of the pearls. So there had been some work made showing that if you take a graft which is really at the border of the mantle, you will improve the color, but you will also, let's say, spread the quality of the pearl between different categories. While if you take the graft from here, which is central graft, you have a majority of pearls of good quality, well, let's say reasonably good, C, well, it's something, but very few very high quality pearls. So you can really play on the graft to get pearls of good quality. but that's not the single parameter. Well, that's it. It was a little bit short. I would have liked to make it a little bit long, but I just tried to make it in 25 minutes.
Okay, there's a question in the chat. It was on the previous topic. Alberto asked the correlation between diagenesis and the variation of the protein profile.
Can it be more precise? Because this is this can be complex.
Yes, it's in the chat. Can you read the chat? It's a quite long.
Chart. Where is the chart?
Is on the top. Just click.
I don't know where I am now.
No, it's easy. Where are you sharing the screen? The same boot on the left. You find the chat.
The left. I try to.
Do you see my screen now?
I can speak to the microphone if you prefer.
This is the chat. You see my.
I will try to. I can. Previous topic, I didn't understand the correlation between diagenesis and I assume it takes place. No, I don't see your screen.
No, but you're reading the chat, no? You were already reading the chat.
Yeah.
I take it out. Okay, sorry. I thought you was reading yours. 1 second.
I just read half of it.
Okay, sorry, I understood. Here is the chat.
Well, I will try to make a shorter. It's a very complex question, actually, and it requires a very complex answer, but I will try to make it short. When we do these diagenetic experiments, we don't say that we try to mimic reality. I mean, maybe somehow it can mimic a little bit what happens when you bury a shell or when you have sediments, above the shell, just accumulating as a pile. What we were really interested in to see, because this was never done before, when we we just wanted to see that first, when you heat a shell, you degrade partly some proteins, some shell proteins. Some of them are really preserved because probably the idea is probably that they are sort of protected by their immediate environment, like the mineral phase. But it's, for the moment, I must say it is difficult to exactly understand what happens, So in the complete list of proteins that we have, when you have when you are dealing with 30, 40, 50 or more different proteins, you know that some will survive, some will not. And some will half survive, and if you hit more, then they will be degraded. So it's difficult to see, for the moment, if, for example, acidic proteins are privilegedly preserved or not. What I saw, for example, and this was a little bit bizarre, but I had the feeling that basic proteins, so proteins with basic isoelectric point, so proteins which are enriched with lysine or arginine, they tend to be preferentially preserved. But that's just a feeling. So it's difficult to, let's say, look at the sequence and say, okay, this one will be this protein will be preserved or not this one. For the moment, we are not yet at that stage, but it will come sooner or later. So is it something? Does it answer your question? I'm not sure, but I tried to make it short.
Okay, thanks.
Frederick, I also have a question.
Mm-hmm.
How does the organic matrix influence the morphology of a shell at the macro scale? There is a relationship between the functionality,
No, we don't know yet,
and the final shape of the shells, for example.
No, this we don't know. What is sure is that the mantle makes the job. I mean, of course, there is for gastropod there is this dissimilarity between the right and left, and the fact that there is a coiling and so on. But for the final shape of what we can see, for example, we see that young Pinanobilis, which is these Mediterranean fan mussels, they have a lot of spines. Well, I can show you here.
Yeah.
I've got some just beside. If you see, do you see?
Yeah.
Yeah. So this animal. This is a young Pinanobilis, 2.5 years old or something like that. So you see a lot of spines on the outside, and these spines are really because the mantle tissue is really making little waves like that, and in one of the waves you will have the secretion of these spines because they are like tubes, like hollow tubes, you know.
Mm-hmm.
So these spines, for example, are really the expression of the fact that the mantle of Pinanobilis has a lot of velocity. But for the shape, the general shape, no, I don't think we have anybody has an explanation for what we see at macro scale about the shape and what we see from the molecular point of view.
Mm-hmm.
I don't think there is a link established.
Okay. And I have another comment. I found interesting your analysis on how many millions of tons of organic matrix is accumulated in the sediment in the ocean.
Potentially, yeah.
Yes, this is also organic matter that is accumulating pollutants. For example, the polyaromatic hydrocarbons, they are accumulated in this organic matrix. So this is also something to think about. This ecosystem services that this calcium carbonate together with the organic part is providing to the environment.
Yeah. I agree, and you know, it's funny because we can think of I don't know for long term to look at past pollution. You know, if some of these let's say shells are preserved in the recent, we can think of the future generation which can probably retrieve some pollution just by looking at what is inside shells. You know?
Yes, exactly.
So I speak about hundreds of years from now.
Yeah. Yes, yes, they can actually check what was the pollutants, the levels, and that's really another story. So there are other questions from the room. Okay, so Frederick, thank you very much. Also, Giuseppe had to leave because he has another meeting. So also from Giuseppe and all the academic board, the colleagues, thank you very much for your lectures.
You are welcome. I hope I didn't make people sleep too much. You know, it's speaking for 4 hours, even with little interruption, can be
It's a challenging, yes, but
it's a challenge for everybody.
For us also, not only for people listening, but also for people speaking. So it's a hard job. Was excellent, very interesting, and I also found interesting your archaeological studies, because we have colleagues in Ravenna with a group of archaeologists that they like to know they have corals. They found corals in the archaeological sites, and they like to know where they are come from. You know, it's a similar question that you help to solve, that you show us today. So when we will talk, I will talk with this colleague about this archaeological project. I will mention maybe we can involve also. You can be involved in this challenging project if it will start.
It's really something, a door that opened. We know I was not so, I mean, how can I say? I was not really prepared to that, but I had this collaboration with Beatrice De Marchi in Torino, and it went very good. I mean, it went really beyond our expectations. So I was very pleased with that.
Actually, the colleagues that introduced me in this archaeological project is Donata Luiselli, is a member of our academic board. So maybe we can, we just have a chat, you know, a preliminary chat. Maybe the next time, if things go on, we'll ask you to join us in the discussion, if you like.
Okay, good.
So thanks again, Frederick. I think the student must go hit something because at 2:30 they restart. So thank you again. We'll keep in touch. Ciao, Frederick.
You're welcome. Ciao, ciao.
Ciao.
Bye.
Bye.